moment, we should start our first session, Nanobiotechnology for Healthcare, first session for today. And our first speaker should be Dr. Vladislav Semenko from the Institute of Scintillation Materials, NASA of Ukraine. Dr. Semenko will present his topic on site. One moment.
Dear colleagues, my report is called Ross Luminescence Centers based on serial nanoparticles with sensitivity improved by FBS addition. And first of all, moment.
Excuse me, can you share a presentation in Zoom? It's real. Okay, thanks.
First of all, what are reactive oxygen species and why we need to detect the content of reactive oxygen species in biological applications. Reactive oxygen species include some free radicals like superoxide anions and hydroxyl radicals, and some more stable molecules such as hydrogen peroxide. And these molecules are formed inside the living cells and play a very important role of signaling molecules, and also they're important in immune response. But when the content of reactive oxygen species is too high, it can be dangerous for the living cell. So how can we detect the content of reactive oxygen species? As for free radicals, the only methods are using of some kind of spin traps or indirectly by interaction with some fluorescent molecules. For hydrogen peroxide, the number of methods is very high. Deuterimetry, electrochemistry, chromatography, and different kinds of optical spectroscopy. And what about using of nanoparticles for this topic? Recently, a number of papers was published about using of different kinds of nanoparticles for detection of reactive species and, for instance, hydrogen peroxide. These nanoparticles range from various carbon materials to doped particles and so on. And in my report, I will show you a new type of luminescent nanoparticle with double action, which can at the same time decrease the content of reactive oxygen species in living cell and to visualize the content of reactive species during the process of this Ross decomposition. And these nanoparticles are well known. These are cerium oxide nanoparticles. They were proposed in about 2005 as unique antioxidant materials. And we have obtained the different nanoparticles with different sizes from 2 to 10 nanometers, which at the same time preserve the characteristic fluoride type structure of cerium oxide. And here you can see more close images them where you can see that nanoparticles are formed mainly by 1-1-1 planes with characteristic, with typical distance between these planes. And so what is our idea? How can the luminescence be used for sensing of hydrogen peroxide? We have observed that cerium oxide nanoparticles have specific cerium 3 plus luminescence. It's so-called 4D5F luminescence. And we have observed that the intensity of this luminescence is highly sensitive to the content of hydrogen peroxide in the water solutions. We have observed that when we increase the concentration of hydrogen peroxide, we have the decrease of intensity of cerium-3 plus luminescence band. And using these dependencies, we can quantify the content of hydrogen peroxide in the solutions. You can see that in the semi-logarithmic scale, the plot is close to linear. As for 2 nanometer nanoparticles, so for 10 nanometer nanoparticles. And these sensors are reversible, so we can use these sensors multiple times by adding new portions of hydrogen peroxide. And at the same time, you can see that the range of the concentration of hydrogen peroxide, which can be detected by the sensors, is in the range of millimoles. It is not very interesting for biological applications where we usually say about micromoles or even nanomoles. So we try to improve the sensitivity of our nanoparticles. And to improve this sensitivity, we have used the different methods. And what was very useful for us is the use of the so-called fetal bovine serum. It's a very well-known biological supplement for the growth of different cell cultures. In our previous studies with our biological group, we have observed that using of fetal bovine serum along with serum oxide nanoparticles prevent their aggregation in the different biological media. You can see the sizes without and with fetal bovine serum. What is the nature of this effect? We think that we have so-called protein corona formed by positively charged globulins and negatively charged albumins, which are the content of fetal bovine serum. And interestingly, we have observed that adding of fetal bovine serum also increases the luminescence intensity of serum oxide nanoparticles. You can see here the luminescence of serum oxide without FBS, the luminescence of FBS alone, and the luminescence of this complex serum oxide plus FBS. You can see that its intensity is much higher than for both these constituents. And we think that this effect can be connected with so-called antenna effect, when the organic molecules of FBS absorb the excitation light and then transfer this light in the form of energy transfer to serum oxide. But this supposition should be checked one more. And what's interesting, we have the increase of luminescence intensity, which provide us the increased sensitivity of our sensors. You can see that while without fetal bovine serum, we can detect only the millimoles. With fetal bovine serum, we can already detect micromoles. So we have an improvement of sensitivity in about 100 or even 1,000 times, which is very interesting for us. Moreover, we also, as with FBS or without FBS, we see these linear dependencies in the semi-logarithmic scale, so it can be used for practical applications for detecting of the content of hydrogen peroxide, as in the technical applications, so in the biological objects. As I have said, FBS also prevents the aggregation of nanoparticles in the different biological fluids. And we have used different biological media, so-called MAN, Hanks, and C199, which usually provide the aggregation because they have a large content of glucose and so on. We see that we have both improvement of stability, lower aggregation of our nanoparticles, and also we can sense the content of hydrogen peroxide, and we have also these linear dependencies. One more moment which can be put forward is that our sensors are reversible. When you use for instance, organic molecules for sensing hydrogen peroxide, they are usually irreversible. They can interact only one time with hydrogen peroxide and so on. And in our case, the sensors are reversible, and this reversibility can be controlled by both temperature and ultraviolet radiation. And you can see that after some time, the sensors in these three cases, the signal of the sensors came to the initial position, which was before hydrogen peroxide addition. So it's also very promising. And now up to conclusions. Conclusions. Nanoseria can be used as hydrogen peroxide sensor in millimolar range. Addition of hydrogen peroxide leads to quenching of cerium-3 plus luminescence due to cerium-3 plus, cerium-4 plus oxidation. Addition of fat or bovine serum allows to widen sufficiently the dynamic range of hydrogen peroxide sensing by nanoseria from millimoles to micromoles, and improve colloidal stability of nanoparticles in biological media. And high sensitivity of FBS-covered nanoseria, as well as their reversibility, photostability, and low aggregation biological media, confirms the high potential of these materials for real-time hydrogen peroxide monitoring in biological objects. So thank you for your attention. I'm ready for your questions.
Thank you for your report. Maybe somebody have questions.
Thank you for this nice presentation. Just have a couple of questions concerning the mechanisms you have mentioned about why the improvement of fluorescence is present and quenching. You said something about possibility of photonic antenna operation for your surfactant, let's say. Does that coincide with somehow with the spectroscopy of these molecules? Because I didn't see the spectra of absorbance or whatever for this FBS.
The problem here is that the absorption spectra of cerium oxide and FBS, they are very close. So it's difficult to separate the luminescence of these two optical centers, really. But we think that we can in some way confirm our supposition.
Okay, but the question is like maybe separately give the spectra just to have an idea. What is the excitation actually wavelength?
Excitation wavelengths, we use 325 nanometers.
Diode, UV diode, yeah?
Yeah.
And the question is, did you consider the most common mechanism of improvement of fluorescence, like surface defects and the coverage of those, let's say, protection of those surface defects by surfactants? This is like very common for nanoparticles.
Yes, when I said about cerium oxide nanoparticles without FBS, it doesn't mean that cerium oxide nanoparticles were not covered. They were covered by saturate. It's also a stabilizer and also protect cerium oxide nanoparticles and improve their luminescence.
Okay.
So,
and then the last question is then, how do you can explain or give idea for the explanation? The FBS is improving your fluorescence, but at the same time, it is improving the efficiency of quenching for the fluorescence. Does it somehow have a general explanation?
Yes, it increases the intensity of the luminescence, and when we have higher intensity, we can observe the minute changes in this intensity. When we have low intensity, we have high noise, and so we cannot detect such small changes when we add.
Okay, I get your point, but actually your curve looks like you have increased improvement of sensitivity, which means that the efficiency of quenching is improving also, which is nice for your application, but the question is why?
It's an interesting question, right?
Okay.
Yeah, we will think about it. Thank you.
Okay, it looks like we have not any questions. Thank you, Vladislav Seminko, for your report. And I invite our next speaker, Dr. Volodymyr Prokopiuk, assessing toxicity of zinc carbonate hydroxide microflakes against L929 cells from Kharkiv National Medical University, Ukraine.
Okay, I'm here. Can I share my screen?
Yes. Please.
Okay. Okay, can you see me? Can you see my presentation?
Yes, it's good.
Okay. Thank you. First of all, I want to thank the organizing committee for the possibility to present the result of our work. Our research is a result of collaboration between three institutions. First of all, it's the Institute of Scintillation Material. Our colleague synthesized zinc carbonate microflakes. And we in Institute for Problem of Cryobiology and Kharkov National Medical Institute investigate the effect of these microflakes on cell lines in vitro. Okay. Right, next slide. Okay, zinc, it's a very important element in biology medicine. First of all, it connected with main fertility and spermatogenesis, and also in with breathing. Zinc take part in carbon hydrase that regulate the dissociation of hemoglobin with oxygen and carbogen in erythrocytes. Also, zinc-dependent protein take part in cell proliferation, cell death, metabolism, DNA repair, antioxidant defense, some cell signaling, transcription factor, and there are a lot of zinc-dependent enzymes. That's why we need a lot of drug with zinc that was a source of zinc for human and animals. But also, always we need to find the balance between efficiency and toxicity of different nanoparticles and macroflakes. Our colleague from Institute of Scintillation Materials synthesized zinc carbon hydroxide microflakes that has two-dimensional structures. It was, as I know, synthesized in collaboration with Institute of Veterinary Medicine for the needs of veterinary and animal husbandry. Our colleague from Veterinary Academy, they already do some research in vivo toxicology experiment, and they found that zinc carbonate hydroxide microflake it's not toxic in mice in orally administration, even in high dose. And the aim of our part of research, it was in vitro test. It was to assess toxicity of zinc carbonate hydroxide microflakes against line 9 to 9. It must be done for understood the toxicity in different form of drug administration. In this slide, you can see our experimental design. First of all, we use line L929, it's a standard cell lines for pharmaceutical research. We grow these cells to monolayer, and after this we add to these cells zinc carbonate microflakes in different concentration. It was 2, 10, 25, 50, and 100 milligram per liter. After 24 hours, we do 6 different assays that reflect 6 different cell functions for understand what cell function can be affected by microflakes. First of all, it was a simple microscopic examination that we can see, first of all, cells integrity, their shapes, adhesion, and confluence of monolayer. Scratch assay, the second pictures, it's assay that reflects cell mobility, cell migration. You can see that the defect has done on the monolayer, and after that, during 24 or 48 hours, we measure the cell migration. MTT assay, it's a very important assay, that a standard assay that reflect mitochondrial function, because MTT reactive goes to the pharmasan in the mitochondria. Another cell function can be measured by neutral red uptake assay. It's panocytosis, ability of cells to accumulate, for example, neutral red. After that, we also do the apoptosis necrosis detection using flow cytometry to find the mechanism of cell death. And we assay also ROS generation. It's a very important assay for nanotoxicology. You have seen in the previous presentation, a lot of nanoparticles induce ROS generation in cells. In this slide, you can see our first assay. First of all, left top pictures, it's a control normal monolayer without microflakes. All cells has similar shapes and 100 confluence of monolayer. The same pictures you can see with microflakes added from 2 milligram per liter and 10 milligram per liter. But when we add 25 milligram per liter of microflakes, pictures changed. Cells lost their adhesion. We have seen a lot of detached cells. Monolayers has maybe 40 or 50% of confluence. As I know from experience, all detached cells, they already die, or these cells in the different stage of apoptosis or necrosis. Those microflake in this concentration are very toxic. The same we see in MTT assay. In the concentration of 25 milligram per liter, microflakes was very toxic, and indices of MTT assay decrease, highly decrease. That means cells lost their mitochondrial function. Without dose-dependent effect, they die. The same we see in the neutral rate uptake test. That means 25% was quite toxic, but no change in the concentration 2 and 10 milligram per liter. The next our assay at scratch test, it's the cell migration. I present this only 3 concentration because in high concentration cells were detached. It's not correct to measure scratch test in attached cells, and we haven't find difference, maybe a little bit increasing of cell migration in concentration 10 milligram per liter, but without statistical difference. It maybe reflects some cells irritation. Also, no difference in proliferation assay. Result of this test is not usual, because usually when we measure some toxicity, we have dose-dependent effect, and first of all, in low concentration, we can see the decreasing of migration and proliferation. In high concentration, when cells begin to die, we can see change in mitochondrial and pancytosic test. Also, we do necrosis apoptosis assay, and you can see that high concentration in high concentration cells die, and we have the pictures of maybe late apoptosis, because cells has inverted membrane with expression on Xen5, and cells accumulate 7AD. Also, we do Ross generation assay in live cells, and we haven't find the Ross generation in live cells. The conclusion that I can say, it's toxic concentration of zinc carbonate hydroxide microflames. Again, zinc line, it's more than 10 milligram per liter. All cellular function are damaged simultaneously by high concentration of zinc carbonate microflames, and cell death occurs through apoptosis without the generation of reactive oxygen species. What can be interesting is that all cells damage, all cell function damage simultaneously. It means this microflake in cell culture damage some basic cell function, maybe like mitochondrial function or protein synthesis. And without energy, all other function can be realized. What can it be usual for us? First of all, it's possibility to coating the biomaterial by this microflakes for antibacterial effect, because microflakes has two-dimensional structure, and the basic cell function, they are similar in eukaryotic and prokaryotic cells. That means it can this material can be very can have antibacterial properties. It can be effective. Maybe these materials can be effective for treatment or prevent some intestinal disease, because these microflakes are not toxic in vivo experiment with high dose of oral administration, and it's toxic for cells without some barriers. Maybe it can be toxic in the intestine for bacteria. And we can use the combination of different types of drug administration. Maybe low dose can be used for intravenous and intramuscular injection, and high dose it can be used for administration orally or covering of some material. Okay, thank you for your attention. Maybe questions.
Thank you for your report. Maybe someone have questions. Okay. Thank you once again.
Thank
We
you.
move to our next speaker.
Finished presentation.
Dr. Anton Tkachenko.
Good morning. I hope you can see and hear me.
Yes.
Share my presentation. Yeah, can you see it?
Yes.
Yeah, okay. So first of all, I would like to express my gratitude for the organizing committee for the opportunity to make my presentation today. And in this talk, I'm on the same nanomaterials, microflakes reported in the previous presentation, but in this talk, I'm going to focus on the hemocompatibility of these nanomaterials, suggesting eryptosis, which is a suicidal cell death mode of erythrocyte, as a toxicological endpoint in nanotoxicological research. Second. Sorry. So in general, toxicity is a huge challenge in the nanotechnology, which significantly prevents the transition of nanomaterials into clinical practice. And hemocompatibility, which studies the effects of nanomaterials on blood and its components, is an important step in the assessment of general toxicity of nanomaterials, because all nanomaterials which can be used as drugs, they enter the bloodstream and are delivered via blood to the target organs. And among the blood cell, erythrocytes are the most abundant, accounting for 99% of all cells in the bloodstream. And they are very convenient tools for the toxicological research because they have quite a simplified structure. They lack organelles, so they don't have nucleus, mitochondria, lysosomes, and other organelles, in order to save more space for hemoglobin, because their major function is to carry oxygen with the help of hemoglobin. And that's why they have a limited set of metabolic and signaling pathways. So all that makes erythrocyte very good model for nanotoxicological research. Currently, unfortunately, there are no standard guidelines for the assessment of hemocompatibility of nanoparticles. However, the assessment of hemolysis is widely used. Hemolysis is a cell death of red blood cells associated with the destruction and release of the intercellular contact. So technically, hemolysis can be considered a necrosis of red blood cells. However, detection of necrosis of hemolysis has certain disadvantages, has certain limitations, because hemolysis belongs to the so-called accidental cell death modes, which occur in response to mechanical damage or physical or chemical damage. So, and this cell death occurs immediately, and it has no signaling pathway, it has no machinery. So we cannot assess the mechanism of action when we study hemolysis. And that is why we cannot influence pharmacologically this process. So there are no pharmacological interventions that can affect this cell death, in contrast to the regulated cell death modes, to which eryptosis belongs. So the regulated cell death modes, they have the machinery, genetically encoded molecular machinery, which can be affected by pharmacological interventions. And that is why, by studying eryptosis, we can also study possible drugs which can be used for eryptosis-related diseases. So in this figure, we have summarized in one of our reviews all cell death modes that occur in erythrocytes. Erythrocytes in blood, they can survive for 120 days. After that, they undergo senescence. During this process, senescence, they start expressing phosphatidylserine molecules, and the expression of protective CD47 molecule reduces on the surface of these cells. And these senescent cells are taken up, they are recognized by macrophages and are eliminated from the bloodstream by macrophages. Also, erythrocytes can undergo two regulated cell death modes, eryptosis and necroptosis. Eryptosis will be the focus of our study. I will talk about it later. As for necroptosis, It is a way of the regulated necrosis. So morphologically, it is associated with the lysis of cells and release of the intracellular content. However, in contrast to necrosis, it's a regulated pathway. So there is a certain machinery inside the erythrocytes that mediate this cell death mode. So, and finally, what is eryptosis? Why it's so important to study? So eryptosis is a regulated cell death mode of erythrocyte, which is quite a counterpart of apoptosis in nucleated cells. However, the signaling pathways differ in apoptosis and eryptosis. As you can see from this figure, the key role in eryptosis is played by elevation of intracellular calcium. So actually, calcium orchestrates the entire eryptotic machinery. It stimulates the action of scramblases, enzymes responsible for the transfer of phosphatidylserine molecules on the surface of the cell, which is a key band in eryptosis. Also, calcium activates calpain, providing degradation of cytoskeleton. Reactive oxygen species are important mediators of eryptosis, and ceramide, which is also responsible for calcium influx and phosphatidylserine externalization. That's why these parameters can be used for the assessment of eryptosis. And here you can see the comparison between eryptosis and hemolysis. So compared to hemolysis, eryptosis has much higher sensitivity. It has a time-saving protocol. It provides good insights into the molecular mechanism in contrast to hemolysis. It provides more precise and accurate data, easily quality control, and higher reproducibility. However, its disadvantage is that it's more expensive. So in this study, we evaluated how zinc carbonate hydroxide microflex affect both these types of cell deaths to study the hemocompatibility of these particles. So here you can see in the upper left corner, you can see that actually dose-dependent effects of these nanoparticles on hemolysis. However, the statistically significant difference was observed only for the highest concentration of microflakes, 500 milligram per liter. As for the osmotic fragility, it was not affected. However, when we assessed eryptosis by traditional methods, by evaluation of the phosphatidylserine externalization by annexin staining, flow cytometry-based annexin staining, we observed activation of eryptosis even at the concentration of 100 milligrams per liter. So a take-home message from this slide is that the sensitivity of eryptosis is much higher than hemolysis. Also, we studied the production of reactive oxygen species in these erythrocytes because they are important messengers, second messengers in eryptotic cell death, and at high concentration, reactive oxygen species production was elevated. What is important? Here you can see also a figure from one of our review papers, which summarizes the redox metabolism of erythrocytes. What is important? The sources of reactive oxygen species in erythrocytes are completely different compared to nucleated cells, because in the nucleated cells, reactive oxygen species are generated by the mitochondria, yes, by the electron transfer chain, while in red blood cells, reactive oxygen species are mainly produced as a result of hemoglobin autooxidation. Yeah, that's why evaluation of eryptosis can provide more insights into the molecular mechanism of nanomaterials, which cannot be studied using nucleated cells. Also, because red blood cells, they scavenge nitric oxide, it's one of the non-canonical functions of erythrocytes, we assessed the side products of NO metabolism, reactive nitrogen species, which can also be toxic like reactive oxygen species and contribute to oxidative stress, yes, mainly like peroxynitride. So here you can clearly see, based on confocal microscopy analysis with probes sensitive to reactive nitrogen species, you can clearly see dose-dependent elevation of reactive nitrogen species production in cells exposed to the microflakes. Surely, on the role of calcium in eryptosis, we assessed the intracellular concentration of calcium. Again, you can clearly see the dose-dependent elevation of the intracellular calcium levels, indicating that really calcium-dependent eryptosis occurs in response to these nanoparticles, microflakes. Like in case of apoptosis, yes, caspases play an important role in eryptosis. However, in apoptosis, activation of like executioner caspase 3 is an indispensable, crucial event. As for eryptosis, the role of caspase 3 is rather supportive, so it's not necessary for eryptosis to occur, for execution of eryptosis. However, in this case, we observed activation of both caspases, caspase 3 and caspase 8, yes, in erythrocytes, suggesting that this eryptosis induced by the microflakes is caspase dependent. So what we can make from this study? That our microflakes that we have studied here, they show quite good hemocompatibility at low concentration. However, at high concentration, they can induce eryptosis in calcium, oxidative stress, and caspase-dependent ways. And what is more important, that eryptosis detection is very good for assessing the hemocompatibility of nanoparticles, because it is more sensitive and can shed some light on the molecular mechanism of the nanomaterials toxicity. Also, I would like to express my colleagues from different Ukrainian institutions who have significantly contributed to this study. And thank you for your attention. Any questions, please?
Thank you for your report. Maybe some questions from our audience. Oh, we have no questions.
Thank you.
Thank you for your report once more time. And we move to our next report. Reconstruction for MRI by autoencoder and periodogram method. Professor Andriy Netreba.
Hello, dear colleagues. Professor Andriy Netreba is my scientific supervisor, and I'm his PhD student, and I will be the one who will make a presentation.
Good. So, launch your presentation, please.
Okay. I hope you can see my presentation.
Yeah, it's good. Take your flow, please.
Thank you. So, dear colleagues, I would like to make a presentation for our research topic, reconstruction for MRI by autoencoder and periodogram method. In medical imaging, such as MRI and CT scans, the signal-to-noise ratio is a significant concern. In CT imaging, increasing of dose of X-ray exposure can reduce noise, but this can be harmful to the patient. In MRI imaging, noise can be caused by patient movement during scanning or low magnetic field intensity. The use of artificial neural networks has greatly facilitated the analysis of medical images with high noise levels. However, these networks typically require a large training dataset and significant training time. In this research, we demonstrate that even with a small training dataset, an autoencoder-based artificial neural network can be effective tool for the noise in medical images. To simplify the task for the neural network, the Daniel periodogram method is employed for preliminary filtering and noise reduction. One of the most common types of noise affecting the quality of MRI images is Gaussian noise. It appears due to the electronic interferences in MRI scanner's receiver circuit and the thermal motion of ions within a patient body. The presence of Gaussian noise can severely degrade the quality of MR images, so they may not provide the quality needed for its interpretation. This noise is modeled by Gaussian distribution with mean and standard deviation sigma. Next slide. In work by Lawrence Marple discussed the classical methods of periodogram estimation, including the in this work discussed the classical methods of spectral estimation, including the periodogram method. It's noted that the periodogram can be effective in reducing noise by averaging the power spectral density over different segments of the data. The periodogram is calculated using the squared sum of Fourier coefficients, as in the following formula. And the coefficients calculation is provided on the slide. However, the standard periodogram had several disadvantages, which led to the development of methods like Daniel periodogram. Daniel observed that standard periodogram could be erratic, particularly with white noise, and suggested averaging the periodogram over neighboring frequencies to reduce fluctuations. An autoencoder is a type of neural network used to learn efficient representation of data, typically for the purpose of dimensionality reduction or feature learning. The architecture consists of two main components, an encoder and a decoder, each composed of layers of neurons. The autoencoder compresses the input data into lower dimensional latent space by passing it through neurons with learned weights and captures essential features while discarding noise. The decoder then reconstructs the input from the latent space using neurons to expand the compressed representation back to the original data dimensions. Convolutional autoencoder in particular is a variation of the autoencoder that is better suited for image data. Unlike the basic autoencoder, which uses fully connected layers, it uses convolutional layers, which are able to preserve spatial relationships in the image reconstruction process. It allows to model. It allows to for the model to understand the spatial patterns and structures in input data. In our convolutional layer-based autoencoder can be utilized for reducing nanoscale structure levels of noise generated for MR, magnetic resonance medical images. The periodogram method, as a good spectral estimator, is proposed for noise variance reducing and autoencoder task simplifications. The diagram you can see on the slide represents a convolutional autoencoder architecture enhanced with a Daniell periodogram method. Initially, the MRI image undergo Daniell periodogram smoothing to reduce noise, and autoencoder then process these smoothed image through the next layers, convolutional layers, to extract essential features by applying filters across the image, then max pooling layers to reduce the spatial dimensions to highlight prominent features, and upsampling layers to increase the resolution and allowing decoder to reconstruct the denoised image accurately. Our research, a small data set of 100 brain images was used. Gaussian noise with different values of the standard deviation was added to the our image to simulate the conditions of noisy MR image. Tests were conducted by varying the size of Daniell periodogram window K. It means the number of points on each side of given frequency used for averaging. The effectiveness of noise reduction process was measured by calculating the Pearson correlation coefficient and peak-to-noise ratio between original noisy image, original autoencoder reconstructed, and original periodogram and autoencoder reconstructed. Further on slide, I will demonstrate the result of the test with values of standard deviation 0.5 and window equals 2. Due to high Pearson correlation coefficient PCC and peak signal to noise PSNR coefficients and much and clarity of visual results. While it's apparent that the reconstructed image does not fully reproduce the original image, we can note a visual reduction in noise, as well as highlighting of brain structures in comparison with the noisy image. This visual observation confirmed by PCC and PSNR coefficients. For the reconstruction process demonstrated in our research, the PCC and PSNR of the image reconstructed with periodogram smoothing and autoencoder is better than simply reconstructed with autoencoder and much better than noisy image. So to sum up, in this research, we investigated the combined approach of Daniell periodogram method and autoencoder neural network as to its capacity as a tool for the Gaussian noise impact alleviation, where it portrays the medical MR imaging. The Daniell periodogram method was applied due to its properties to provide initial noise reduction in the frequency domain. The convolutional autoencoder with its ability to preserve spatial relationships was then utilized to further enhance the MR image quality. The denoising result achieved through the integration of a convolutional autoencoder and Daniell periodogram method were tested with artificially added Gaussian noise, and the result can be observed visually and by Pearson correlation coefficient, also by peak noise to signal to noise ratio. The original reconstructed image PCC increased 4 times compared to the original noisy image, and PSNR increased approximately in 2.5 times. Combination of these two methods shows potential for preserving crucial anatomical structures in noisy MR image and does not require large training data sets. So thank you for your attention.
Thank you for your report. Some question to our reporter. Okay, no questions. Thank you once more.
Thank you.
Time. And now we have on-site speaker, Professor Dmytro Snishko, with his report, Pulse Technique for Nanoelectrochemical and Electrochemiluminescent Analysis in Biomedical Applications from Kharkiv National University of Radioelectronics, Ukraine. Please take your floor.
Good morning. So I represent today two groups. My university, Kharkiv National University of Radioelectronics, and Changchun Institute of Applied Chemistry. These are our colleagues, were me and my colleague Zholdov to do some part of this work. And today we will talk about pulse technique for nanoelectrochemical and electrochemiluminescence analysis in biomedical applications. Electrogenerated chemiluminescence, it's enough interesting method that open possibility for nano analysis and nanoelectrochemistry. Generally, this method based on radical recombinations, which associated with light emission. And this light emission enough interesting key for analysis of content different solutions. In many cases, radical recombinations change it by reaction with correctance. So one ion generated by electrochemical process and other can be chemically available in solutions. In this case, we have correctant ECL reactions. Most known reactions with halide complex of ruthenium, which you can see here, this one, and mostly used in 99% in analysis. But our group tried to expand this possibility for ECL analysis and look how to apply ECL for nanoelectrochemical applications. Today I will talk about pulse technique. Why is important to change DC excitation of reaction to pulse technique? This enough simple answer if look how to change involved solution in electrochemical process. When we reduce time, we reduce also diffusion time for species which involve in reactions. So when we reduce down to microsecond regime for our electrolysis, we reduce to nanosize layer which close to electrode which involve in reaction. So we can examine probe in nanolayer, and sometimes we also possible reduce to size of molecules. But also, if we talk about specific techniques as like potential sweep voltammetry, we need also possibility for features of our instrumentations which available for fast polarization. And this one enough important that sometimes doesn't realize in general potential states. So in our laboratory, we use as like standard potential state, for example, Autolab PG state 128, but also develop specific instrumentation for fast technique, and we'll see that this one will be usable for next applications. So our measurement system use fast potential state and also instrumentation for a fast analysis of optical emissions. This one pulsar counter. So if we talk about ECL and the advantage of this method, we detect light, and this one most important advantage reference to the electrochemical methods. Because if we talk about electrochemistry, frequently we use for analysis potential state. And most potential state based on using of operation amplifiers which can work begin from thousands of electrons. So our acts of electron transfer in reactions we can examine only when we have thousands of acts of reaction. But when we talk about photons, modern instrumentations available to detect separate photons. Yes, of course, not each one photon, but enough close to this one. So when we talk about photon counting regimes for sensors, we can step down to detect separate acts of our reactions. So this one enough important to and open way for work with single molecules detections, single particle detections, and so on. So our instrumentations include, yes, of course, instrumentation for electrolysis, photomultiplier tube. If we talk about fast experiment, we use radio engineer instrumentations, generator, oscillograph for fast signal registration. Yes, of course, in some cases we use barbotage for electrochemical cells. So, and this one, yes, of course, inside the light-proof box, because we try to detect separate molecules to improve our limit of detection. And yes, of course, if talk about Helat complex of Ruteni, it's not very fast correctant reaction because includes many steps, and as a result, we do not have fast response. So for our laboratory was interesting try to use something new, and in our laboratory was found correctant which you see tetraphenyl borate, which enough interesting correctant, absolutely other than we have different tripropyl amine for ruthenium Helat complex, and this one processes how it can be involved in ECL and generations. So we try to examine mechanism of ECL reactions for this correctant, and this one you see electrolysis response for this corrector. So if we back to ECL mechanism, you can see frequently we records as electrochemical response as ECL response. And if you look in case present of different component in solution, why is chemical electrochemical detection so maybe raw, not so precise, because we work again with multiple number of electrons which produce in electrolysis, and in solution can be present many components which can involve in this electrolysis. But when we talk about specific reaction, ECL reaction, separate photons, this one mark that in solution present analyte. And as you see, sometimes electrochemical response are not associated with, directly are not associated with ECL response. So we do not have cross connection, because this one you see correctant oxidation. And here ECL reaction. So this one absolutely separate peaks, and we obtain additional information in optical channel. This one advantage of ECL. And for us was interesting also, as we from biomedical engineer department, application of our correctant for detection biological important molecules. And example enough interesting, this one tryptophan. Tryptophan luminescent amino acid, and as a result we try to wait that it can be involved in ECL reaction, and with new correctant it can be done. So you see how to change response, ECL response in reaction with correctant triphenyl borate, tetraphenyl borate, sorry, tetraphenyl borate. And these one conditions. So as excitation, we use pulse enough short time for to obtain. Why is we use pulse? Because this one at first work enough fast, this one correctant, and other situation that disadvantage of this correctant that it accumulates on the surface of electrode, and the electrode surface degrades during analysis. So pulse technique to show more more better than other technique for analysis of this correctant. But degradation not so fast, and we have enough good repeativity that you can see how to change pulse numbers in different conditions and different concentration correctant. So it's possible to find conditions when we do not have too much degradations and enough good repeativity in our experiment. If need to examine several times or use in several experiment electrode. But also for us was interesting to use photon counting regimes, as you see here. This one responds with high time resolutions, and this one to show how to change ECL response during the pulse. So analysis at low concentration to show that peak is less reproducive than integral square of response. Under this one luminescence curve. So integration signal more better represent concentration of reagent at pulse technique for this one pair, tryptophan tetraphenyl borate. Also, when we develop a method for analysis, we examine conditions at which one better response, and as for biological samples enough important pH of solutions, we also analyze in wide range response, find optimal conditions. Enough important was to verify what is the source of luminescence, because tetraphenyl borate also at specific condition can be exhibit emission in electrochemical process. So signal enough low at ECL reaction, and to use general spectrometer is impossible. So we use numerous spectral filters, optical filters to reconstruct and examine who is agents of luminescence in ECL reactions, and we verify that the peak of our agent enough close to emission of tryptophan. That enough important. Also, because in biological samples we have multiples other amino acids, we try to examine specificity of our method, and for this reason we examine other amino acids. But as you see, we have enough specific situation for ECL and intensity of chemiluminescence reactions, electrochemiluminescence reaction, we can see only in case with tetraphenyl borate and tryptophan. And this one enough important. So adding of alanine, histidine, tyrosine, or cysteine doesn't change significantly on the ECL reaction. Addition measurements and experiments to show us that important role in ECL production and emission play indole group play significant role, and as a result compounds with this one group can be potential substance as analyte for develop next methods based on tetraphenyl borate correctant using. So in this one report, I try to show importance pulse technique and development electrogenerated chemiluminescence method to analysis and to show advantages of this method in comparison with electrochemical methods, and try to show difference with maybe other luminescence method, because this one open new possibilities, add additional information in analysis, and enough controllable in experimental conditions method. Thank you for your attention. This work supported by VIEW projects at, yes, of course, bilateral Ukrainian-China project, government support by Ministry of Education and Science of Ukraine, and Chinese Academy. We was with colleague as invited scientist in Changchun University. This group with us. Yes, of course, my colleague. I forget where me. And yes, of course, I want to congratulate everyone with today celebration of 33rd Independence Day of Ukraine. So thank you for your attention. I will be ready to answer on your questions.
Thank you for your report. Maybe some questions from our audience online, on site. No one. Thank you once again.
Thank you. Thank you for organizers.
And our next speaker, Professor Sergei Shulga.
Yes, I'm here.
With his report, Features of Creating a Liposomal Form of a MicroRNA Curcumin Complex, Institute of Food Biotechnology and Genomics, National Academy of Science of Ukraine.
Yeah, but I send you my presentation. Can you show the presentation?
1 minute.
Yesterday for the organized committee.
1 moment, please.
Okay.
Okay. Few seconds and we launch your presentation.
Yeah, thank you. First of all, thank you for the invitation and the opportunity to show our work. Feature for the creation a liposome form of a microRNA curcumin complex. Next, please. The aim, the development of a new drug aerosol lipid target system containing curcumin and microRNA, maybe not really drug, maybe prodrug for the treatment of the neurodegenerative disease. Determination of anti-inflammatory and anti-amyloid activity of the drug. It's efficiently in relation to the dose of the duration of action and after action in vivo. Sorry. Next, please. This picture demonstrates or illustrates the progressive of Alzheimer's disease. On the left, you can see the normal or healthy neuron with a normal microtube or with tau protein and beta amyloid monomers which create from the so-called amyloid precursor peptide, APP. Maybe on the next slide I explain more exactly. This protein cut with the 3 secretase for the amyloid monomers. Then these monomers form oligomers, and oligomers form plug and then plug, fibril and plug. On the right hand, you can see the neurodegeneration which destroyed the microtube with the super phosphorylation and destroyed tau protein. And then the plug block the signal during the synapse and axon. Next, please. This is schematic structure depicting beta amyloid in the main aggregation component of amyloid plaque in the brain. Well, you see on the left part of these pictures, this is usually a form of non-amyloid organic protein, so-called SAPP alpha. And on the right hand, you can see how to prepare the amyloid organic situation when appear the beta amyloid peptide, so-called AB. The beta secretase cleavage for the APP, then work gamma secretase, and we receive the beta amyloid peptide with the long 140 and 142. This is beta amyloid peptide has a high tendency to accumulate oligomers, aggregates, and forms amyloid scenes plaque, resulting in the substance alternation in the Alzheimer's disease. Next, please. We try to prepare the drug delivery system. Looks like the liposomes. The liposomes was built or created from the lecithin, which we receive from the sunflower lecithin. This is a dry powder lecithin, which we receive from the phosphatide concentrate of sunflower oil. As usually we work when we extract the phospholipids from the phosphatide concentrate, we receive the dry powder lecithin. As usually we work with lecithin, which consists phosphatidylcholine, phosphatidylethanolamine, phosphatidylinositol, and lysoform, lysophosphatidylcholine. Next, please. Yeah. We usually work and try to prepare the liposomes form with two methods. First method is so-called extrusion through the membrane with a pore step-by-step, 200 nanometers, 100 nanometers, and 50 nanometers. So we receive the liposomes with dimension diameter 50-55 nanometers. And the next method, sonication, we receive the liposome dimension of 100 or 120 nanometers. We work with two maybe pro-drug or two components of liposomes which include in liposomes. First of all, curcumin. Curcumin, well known from the Asian medicine from India or China, and very popular in India. And now we really understand that curcumin very popular for the food industry, even in Ukraine, as a color. But nevertheless, it's possible to, you can see that the form of curcumin depends in various pH system. Next component is a really microRNA. MicroRNA looks like the pin. So we can see the double pin and one layer. It consists with the 20 20. Yes, 20. You see from the 47 up to the nucleotides. Next, please. When we obtain the liposomes, this picture demonstrates the liposome form of sunflower with a curcumin, which we receive by the atomic force microscope. This is on the left. These are the 3D pictures, and on the right in the plane. Next, please. How we prepare the model of the Alzheimer's disease. Alzheimer's disease we prepare with intrahippocamp injection, beta amyloid 40 human to experiment rats and in a stereotaxis setup. Well, you see, this is a very tricky situation, how we can really penetrate the beta amyloid 40 to the hippocamp. It's you see, it's necessary work very hard that to prepare the injection directly to the hippocamp. Next, please. Well, next step, we try to investigate the situation with the liposomes and how distribute the curcumin and microRNA in liposome. First of all, as usually, we obtain that curcumin penetrate to the bilayer of liposomes in the phospholipids bilayer. And microRNA, they really penetrate to the water core of liposomes. But for us, it's really very interesting, very surprised that we receive the pictures and the results that curcumin penetrate to the water core together with microRNA. You see, this is the phase contrast with curcumin, green color. This is separately situation when the microRNA penetrate to the water color in the water core, the red color, and this is together curcumin and microRNA. This is really for us very surprised that maybe we have the some interaction with curcumin and microRNA. Next, please. This is maybe not so good, but nevertheless, it's possible to demonstrate once more the situation of exactly how it distributes the curcumin and microRNA and complex microRNA and curcumin in liposomes. Next, please. And this is composite image projection. You see with 39 individual slices on the XY plane. This is well known, the situation with the phospholipids around in the green color you see, and curcumin and microRNA. Next, please. This is the same situation, how the projection of really localization microRNA with CY5. This is a special red color, so we received the red answer in the picture. Next, please. And this is a three-dimension reconstruction of reveal microRNA localization in the liposome thickness. You see it possible demonstrate in the you see in the thickness. Next, please. This is you see the small conclusion of our investigation, but nevertheless, we at the same time try to investigate the you see so-called pro-inflammation of interleukin, such as interleukin-1 beta and interleukin-6. The volume of these interleukin down with interaction with curcumin and you see microRNA more than 60%. So it's possible that this you see this so-called prodrug curcumin and microRNA work in the right way for the protect the first step of you see appear of Alzheimer's disease. This is the small conclusion. And next, please. This is the research staff participant in the present research. Next, please. Thank you for your attention. Thank you.
Thank you for your report. Please, have anybody some questions? No one has questions. Thank you one more time.
Thank you.
Invite our next speaker, Dr. Oksana Harchenko, with report poly-to-oxazolines-based lipid nanocapsules as a PEG-free vehicle in nano drug delivery from University of Angers, France.
Okay, thank you so much for introducing me. One moment, I'm sharing my screen. Let me know if you see my screen.
Please launch your presentation in a full screen mode.
Not full size.
Not full size.
One moment. Now how it looks.
Oh, okay. Please take your flow.
Okay, thank you so much. Hello, everyone. Thanks a lot for this possibility to present some part of my scientific work. And today I will talk about poly-to-oxazolines based on lipid nanocapsules, a spec-free vehicle in nano drug delivery. First of all, water-soluble, biocompatible polymers are important building blocks in a drug delivery formulation. From micelles to drug polymer conjugates. Polyethylene glycol is often considered the gold standard for biocompatible and inert polymers used in drug delivery formulation. PEG has been widely used in biomedical application because it imparts like stills properties for some drug delivery system. Although PEG has been considered bio-inert, recently some serum antibodies antibodies against PEG have been detected in some patients that were treated with PEG conjugates. And moreover, synthesis of PEG has some drawbacks. For example, it can forms peroxides. Polyoxazolines are versatile polymer class which have been identified as a potential PEG replacement due to their biocompatible properties. And polyoxazoline has some advantages compared to PEG with regard to their synthesis, solubility, high drug loading, immunogenicity, and low toxicity. And in addition, in vitro studies with polyethyloxazoline and polyethyloxazoline copolymers were reported to be biocompatible even at high concentrations than polyethylene glycol. The synthesis of polyoxazoline by cationic ring component polymerization opens up numerous opportunities. First of all, the functionalization of POX can be performed in alpha and in omega position that leads to obtaining polyoxazolines with different targeting groups, which can be interesting for biomedical application. And additional functionalization can be on a side chain structure of these polymers. Polyoxazolines are being investigated in a variety of pharmaceutical and medicinal application, for example, as drug polymer conjugates, micelles, and lipid nanoparticles. In this work, I would like to present design of amphiphilic polyoxazolines, which can be used as a surfactants for nanoparticles formulation. And here we have shown polyoxazoline-based alternative lipid nanocapsules, which can be alternative for lipid nanocapsules, pegylated ones. Among the wide application of LNCs, they have been applied for delivery various types of active components, especially for treatment of cancer or imaging. And I just wanted to say that lipid nanocapsules were developed at MINT laboratory where I work now. And this laboratory is focused on nanomedicine application. The chemical versatility of cationic ring component polymerization enables the introduction of lipid motifs via several synthetic pathways as the initiator and termination method. The amphiphilic polyoxazoline surfactants were synthesized through CROP, or cationic ring component polymerization of 2-methyl-2-oxazoline by using 1-iodohexadecan as initiator in this method, or steric oleic palmitic acids as terminators. Although molecular weights and polydispersity indexes were characterized by SEC, and for all polymers, molecular weights were obtained as expected to theoretical ones. Lipid nanocapsule consists of oil core formed by labrafag and outer shell of surfactants. In our case, it's polyoxazoline surfactants and co-surfactants lipoate S100. And the phase inversion temperature method was used for LNCs formulation, which can based on mixing all components at the beginning, and then 3 heating cooling cycles were performed, followed the sonication step, and after 1 heating cooling step, emulsion was diluted by rapidly addition cold water that leads to obtaining lipid nanocapsules. And plenty of formulations were done to find optimal conditions to obtain exactly epox lipid nanocapsule, because it's not the same procedure compared to classical method of to obtain like pegylated lipid nanocapsules. The hydrodynamic diameter of this epox lipid nanocapsule was in the 130 nanometers with PDI around 0.157, and zeta potential was in the range -1120, for example. And as one of the application of polyoxazole-based LNCs, it's imaging thanks to FRET effect. FRET, or fluorescence resonance energy transfer, it's a mechanism of energy transfer that is widely utilized in biological experiments. And FRET can be like ideal pharmacokinetics of lipid nanocapsules as nanocarriers before clinical application. In the present state, we co-encapsulated 2 fluorophores, DID-TPB and ITPB, able to interact by FRET in a lipid core of lipid nanocapsule. And it should be noted that these fluorophores are not commercial ones, and they were synthesized in our lab according to known techniques. And lipid nanocapsules, the formulated LNCs with FRET dyes were characterized by DLS, and hydrodynamic diameter is found to be in the range of 19 nanometers with PDI 0.235, and zeta potential was in the range of 14 millivolt. And it's well known that FRET can only occur at a very close distance, around 10 nanometers. And FRET relies on an interaction between 2 encapsulated dyes, which is more precise for monitoring that a single encapsulated dye system. And here it's shown a fluorescence spectrum of FRET epox LNCs. And as we can see, it's like confirmation of FRET effect, because all energies that was absorbed from DII was transferred to DID, and here we can see intense signal of DID TPB dye encapsulated in LNC. And also this formulation was tested after centrifugation, and we can see this dashed line that this just in intensity of fluorescence just confirmed us that it still exists FRET in LNCs after centrifugation formulation. And for practicality, efficiency of FRET is simplified by correlating to the donor and acceptor intensity, giving a practical parameter called proximity. as it's shown in the the proximity ratio of FRET LNC's formulation was 0.94. That is optimal FRET signal, and it demonstrates the full integrity of lipid nanocapsule. And as other application for POX by drug delivery system. So we've done formulation which consists of a photoactive agent for photodynamic therapy. It is a photosynthesizer. This PDI or pyrilendiamide, one as PDI, nano-tunated pyrilendiamide, is known as efficient photosynthesizer. And this highly hydrophobic compound was successfully encapsulated in a POX-based lipid nanocapsule, and this formulation characterized with hydrodynamic diameter 180 nanometers with PDI 0.198. And I want to go back to the synthesis of these pyrilendiamides. And this of pyrilendiamides were synthesized according to two methods. One method is that Levson reagent can be used for synthesis of tunated PDI, but in this case, this method has the main drawback. It's low yields of targeted products, less than 10% for each tunated derivative. And in our group, it was developed a new method of tunation pyrilendiamides, and it was shown that Curfews reagent or combination this reagent LSP4S10 and HMDSO can improve yields of the reaction and can go to rapid conversion to tunated derivatives. And in a case of PDI with alkyl substituent, the new 3 and 4S PDI were synthesized first time. That was not described previously. In the case of 4S PDI, we were able to increase yields to 32% compared to known in the literature 9%. And also, as I told, that 1S PDI can be used as a photosynthesizer. It is really important to know quantum yield of singlet oxygen generation. And based on experimental result, it was calculated quantum yield for 1S PDI, and it's found to be 0.94. That is much higher compared to known photosynthesizers, for example, chlorine E6. That quantum yield for chlorine E6 is 0.7. And also, as additional application, we are going to do, in our perspective, we are going to encapsulate 4S PDI in our POX-based LNCs. And 4S PDI can be used as a photothermal agent and for photothermal therapy. And details of this new method of synthesis you can find in an article that was published this year. And to conclude this work, I would say that new formulations of POX-based LNCs were done, and we successfully encapsulated FRET dyes in our POX-based LNCs,
Thank you for your report. Maybe some questions. Okay, we have no questions. Thank you one more time. And I want to invite our next speaker, Professor Serhiy Kurta, with report Nanostructure and Biochemistry of Vegetable Squalene and Squalene for Cosmetics.
I congratulate the participants of the 12th International Conference, Nanotechnologies and Nanomaterials. From the Department Chemistry of the Prikарpat Nation University in Ivano-Frankivsk. And I have say that I would like to express our special gratitude to our sponsor, the Austrian company Shark Product, two representatives of which, namely the Anastasia Zhadan and Tina Turner, are a present of our conference. And thank you for Mikhail Gorst and representative of this company. Okay. This key word of our work was the natural hydrocarbons, squalene and squalane, and method of their production and properties. Squalane and squalene are a natural oil. They are extracted from the shark liver or from the amaranth seed. On the side, you can see manufacturing of this product from the Japan and United States of America of our slide. But you must know the approximately 3,000 sharks are required to produce 100 kilograms squalene. Due to environment concern, our resource such as amaranth and oils, rice and others have been the commercial and the 20 and 24 years supplied to the 40% of total squalene and squalane industry in the world. Okay, squalane and squalene are a hexaoligomer of isoprene, is the organic unsaturation compounds with the formula number 1 and squalane formula number 2. It's this saturation product. Hexa saturation squalane formula number 2. Formula number 2 have the 6 double unsaturation and saturation in intermediate links in the biosynthesis of cholesterol, beyond to the group of the carotenoids. The names come from the Latin squalaus. Squalaus is this shark. Okay, on this slide, we have physicochemical properties of squalane and squalene. In the table shows the content in the natural raw product, whereas is less than 1%. But shark liver, its content does not exceed 1 or 2%. And the amaranth oil, squalane, can reach to the 6 or 8%. This is squalane on the shark and amaranth oil. That's slide number 5. Okay, preparation which they include amaranth and squalane are useful in both medicine and cosmetology, which recent hide does of the radioactive radiation. And the addition is effectively restorative of the skin, which is why in the considered and the revolutionary drug in cosmetology. The composition of the amaranth oil is present in the table number 2. This is contain saturation and unsaturation esters and fatty acids, for example, olein, palmitin, stearin, and linoleic acid and glycerin. On this slide, we can see in this slide, we can see the reaction which we can place is very interesting of our skin. Is a very property of squalane. It is able to react with the water and the peroxide water molecules and release atom the molecule oxygen, turning into the squalane according to the reaction 3 and 4. The squalane can reserve as a resource of oxygen, which is necessary for the human body. Research of the conducted for many scientists of the world has allowed establishment the anti-cancerogen, antimicrobial, and fungicidal effect. Therefore, squalane and squalene are widely used of the medicine and perfumery, pharmaceutical, bakery, and others. At the chemical department and the Faculty of the Natural Science and Vasil Stefanik University in Ivano-Frankovsk, in the botanic age of our university, we conducted applied studies of the various technological methods of the separation and the paraffinization squalane and squalene from amaranth oil. On this slide, we can see amaranth, which we roast of our botanic age, and we can see some amaranth oil of this slide. The two various amaranth oil, violet red and green red, the two sort of amaranth. We can rise of our botanic that in 20-24 years in Prikарpat Nation University. Amaranth oil, which we recently used, obtained the squalane oil and the clean and liquid for the light dark yellow. and but they're very expensive. But 1 liter of amaranth oil is cost of the 2,000 Ukrainian hryvnia. And this slide, we can see the amaranth, which we roast of our botanic site, and one red color and the green color. Two where the amaranth colored red and yellow. We are rest on the botanic garden and the Prikarpat Nation University, and this year. Okay, the method processed by us only the high paraffinic and olefinic hydrocarbons, which their carbon content reach the carbon 30 and hydrogen 62. At the same time, the squalane loss decrease, and we are able to separate the 30% squalane from amaranth oil. Through the amount unsaturation squalane, one carbon 30 and 52 decrease to the 1, and the amount the saturation squalane increasing to the 8%. According to the reaction, slide number 6. Number 6. Okay, and the sorry, my English is not so well. Method number 2, we have patented and new method of extracting squalane reactive of the water and extracting squalane from the peroxide hydrogen. This is a very valuable property of unsaturation squalane is that only surfaced on the skin in the under and right condition, react with water and hydrogen peroxide from the human body, and at the same time released atom and molecular oxygen transformation and hydrogenation into unsaturation squalane according number 2 of this reaction. Qualification indicator amaranth oil of this table, we can see the obtained extraction of amaranth seed. Okay, and we analysis of our product, and we can see that some pieces of our samples of amaranth and amaranth squalane, and at the same time the completion of the composition of this loss 100%, and maybe so some very extraction index. Oh, this is a reaction. You can see with the first reaction, the reaction with water with squalane, and we obtain this unsaturation squalane to unsaturation squalane. And oxygen is the old of the air. Is oxygen is very reaction of our aim. This is apparatus for our experiments, and this is infrared spectra of our products and intersection of our substances. This is a result of DTR detection of our products. And this is same amaranth oils in indication of spectrophotometry method, and we obtained some very interesting result which we can do some very interesting research. And liquid chromatography, as we can see of this slide. And in the conclusion, we can say, in the way, it is possible to establish a production of product squalane and squalene in Ukraine as a cosmetic and pharmaceutical means of the protection human skin, including from the wound and from during military operation. The main conclusion from our work that we have found as a new technological method from the isolating squalane and squalene from the amaranth oil and rectification them to the 55 and 95 years purified from the raw material amaranth oil. And thank you for attention to me. And we would like to express our special gratitude to our sponsor, the Austrian company Shark, two representatives of which, namely Anastasia Zhadan and Tina Terman, are present of our conference. And thank you for Mikhail Grotz for present of our conference. And I listen your question to me.
Thank you very much. Do we have any question?
Please.
Yes, we have one question.
Sure.
One
Please.
moment.
One question.
I see.
May I ask question, Shulga? Well, you see, you talk about the nanostructure of squalene and squalane. Can you explain more detail? What does it mean, the nanostructure? I cannot saw this nanostructure. This first. And did you really receive squalene and squalane in what form? This is the powder or this is a liquid? Exactly squalene and squalane. Please.
Okay. Thank you for attention and thank you for questions. Squalene is liquid and saturation, but squalane is some maybe so some liquid on the solid, but maybe so paraffin, maybe so nearly paraffin, and we can extraction squalene, but some of our method produce some we obtain the squalane, but squalene it's hydration to the water, and we obtain the squalane. And in our sample, we obtain the 50-50 squalene and squalane of our product. And I can have of some product of extraction of amaranth oil. This is liquid, maybe so Zol, and this is squalene for the some maybe so liquid on the on the solid. This is example.
Okay, thank you for your answer. We have one more question in our hall.
Thank you for your presentation. I have one question. I can see you synthesized squalane from squalene. And my question is there a way to synthesize more rare squalene from squalane?
But we don't synthesize squalene on squalane. We extraction method from the amaranth oil and others oil and obtain the squalene and squalane. But on the product of this method, we can obtain squalene and squalane, but sometimes we obtain this squalane and maybe so 80%. But squalene is very unstable for a method of our obtain.
Thank you. Thank you very much.
Thank you very much.
And the last question, please.
You were speaking about the reaction in which squalane reacted with water or hydrogen peroxide with the release of oxygen.
Yes,
It is something very, it is a very unusual, very surprising type of reaction concerning unsaturated hydrocarbons. Yes,
Yes,
exactly this
yes, I
method.
know.
Yes.
You are right, but not exactly. and our skin, we sometimes the squalene produce of our skin in the liquid and obtain of our skin and some hydration was water and obtain with some solid phase of our skin, and this reaction in the explaining of our squalene to the transformation on squalene of our skin. But this process is not chemical. It's very difficult to understand to me, but maybe some in future we can explain of this method with the biological and other scientists.
So thank you very much once again for your topic and for answering the questions. And we will move forward according to our program. The next speaker should be PhD student Volodymyr Hrin from the National Academy of Science of Ukraine Institute of Physics. Volodymyr Hrin, please take your floor.
Dear colleagues, good morning. Today I want to talk about features of the element production for enhancing the effects of resonance reflection and Raman scattering. This work were performed by 3 work groups from the Institute of Physics of the National Academy of Sciences, from the Fraunhofer Institute for Applied Polymer Research in Germany, and Lviv Polytechnic University in Ukraine. So we work with photocuring materials, photocomposites, and this is very perspective materials that allow us to create different types of photonic elements. Such of these elements were used for the sensing application and for the enhancing of signals. Nowadays, such structures like sensors made in the relief materials by using, for example, electron beam processing, but this methods is such hard to provide. And for the other side, resonances on these structures may be some wider that decrease the sensitivity of the detectors. So what was our idea? We want to use photopolymer materials for creating such resonant structures for sensing applications, and by using holographic lithography, we can easy to structure to to structure the structure. Okay, and our theoretical analysis shown that we can increase sensitivity of this element. So we want to try to make testing samples for sensing application in photopolymers. So what is this is resonance Wave-Git structure. When we have some layer with periodic distribution, for example, nanoparticles. We use nanoparticles because they increase modulation of the refractive index of this layer, and we need to make a thin layer. This layer will be like waveguide, and then we have interesting effect. Waveguide wave-guided beam and diffracted beam join into the resonance. And in the reflectance or transmittance spectra, we can show resonance peak. When we put some liquids or analytes on this structure, we can show shift of this peak. And we can detect our change of refractive index. So our theoretical group from the Lviv Polytechnic University, using rigorous coupled wave analysis method, calculated what thickness of this waveguide layer we need to make. So if we have about 1 microns of this layer, when we have only one waveguide mode. For 2 microns layer, this is 3 modes, and for 5 microns layer, we have 6 modes. It's not good. We need to make thin layer, thin photosensitive layer with thickness about 1-2 microns. So we try to make it by 3 methods. By spin coating, by filling cells, and by pressing photocomposites between two substrates. All thicknesses were measured by Dektak spectrometer in Institute of Applied Polymer Research in Germany, or by microinterferometer. So the first method, by using spin coater, we have some difficulties when we try to put another substrate because of the edges of the glass. We have many air between the substrates and the polymer layer. Why it is why it is so difficult for us? When we have air, polymerization process doesn't provide. So we need to use, for example, a vacuum chamber or chamber with inert gas atmosphere. We have a very good uniformity of layers, but we can make easy holographic recording for structuring this polymer layer. The second step, we fill the cell between two substrates. And we obtain good results for this method. Okay. We obtain good result for this method, but layer is some thicker than we need. The next our method is pressing method by using Dektak, sorry, not Dektak, by using a press handle. And we get very good results for this method. For thicknesses, depending on load, we can control our thickness. We have a nice uniformity of layer, small wedge, but it is okay. So here you can see the layer surface. As you can see, between two substrate polymerization, it is good. It homogeneous. We have thicknesses about 1 to 2 microns, and with spin coated and vacuum, we have such less good thicknesses. So we need, as I previously said, we need to organize our layer to make a periodic structure. We using a holographic recording method, a scheme of experimental setup you see on the slide, and after that we have diffraction grating with a good parameter, about 90% diffraction, high modulation for refractive indexes. We can control thicknesses and we can control period of these structures. So the next step, it was testing such periodic structures as a sensitive element. We make a liquid cell, which we put on the spectrometer setup, and fill it with the different liquids. And we see good dependencies, which provide us with good sensitivities, about more than 100 nanometers per refractive index unit, and with high quality factor, more than 50,000. So I can conclude that testing 3 different methods, spin coating, filling cells, and pressing. The best way was the pressing method that can control our thicknesses. We can provide a holographic recording in this sample. And after that, we have a high Q factor structures with a high sensitivity. Moreover, as you see on this slide, our full wide on the half maximum spectral wide is very narrow, so it limited by the spectral equipment. So we have only about 12% of reflectance, but theoretical analysis show us about 100% of reflection. So we have equipment limits, unfortunately. For this work, we prepare 5 publications, and this work was supported by the NATO project, multi-year program Science for Peace and Security, and the targeted program for fundamental research of Ukraine. This is our team who work on this project. Thank you for attention, and I'm waiting for your question.
Thank you very much for the presentation, and we already have one question in our conference hall.
Thank you for the talk. Am I correct that you have some like a photonic crystal, so the stack of polymer and substrate, yeah, with a thickness about 1 micron? Or not? This is the whole structure.
As I clearly understand you, you're asking, is it photonic crystals or?
So first of all, the structure consists of 2 types, right, of layers, substrate and polymer, right? Periodic structure with the thickness of the layer is about 1 micron, right?
We have 2 substrate. And we fill a photosensitive composite between this substrate.
So this is homogeneous or not? Because first you said homogeneous, and you show some like a grating or pattern.
In liquid phase, it homogeneous. It contains from different types of monomers and neutral component nanoparticles.
Okay, so then what is the origin of this resonance?
Origin of this resonance is the interaction of diffracted beam and incidence beam and wave-guided beam in the wave-guided layer. So this beam interacts and resonant.
Okay, thank you very much for the discussion. Do we have any more questions? No more question. So once again, thank you, Mr. Volodymyr Hrin. And we'll move forward to our next speaker, PhD student Pavlo Hort from the Taras Shevchenko National University of Kyiv, Ukraine.
Yes, thank you. I will now start my presentation.
Okay, please. Now we can see your presentation. You can start.
I can see my presentation on the screen. Okay, so then I will begin. Good morning, dear audience. I'm grateful to have the ability to present you our latest work. My name is Pavlo Holt, and I would like to present you our work about the study of photodynamic efficiency of hybrid star-like anionic polyacrylamide polymer, gold nanoparticles, and thermopropane photosynthesizing nanosystem. The idea of our work is to create a new system for the photodynamic therapy, but to do this, I would like to first say a few words about what the photodynamic therapy even is. It's a quite novel and promising non-invasive treatment method that is used for a wide range of diseases, but our systems that we have been developing is mostly targeted on the cancer treatment. Since the first successful clinical trials of the photodynamic therapy, there was a number of studies targeted on the increase of efficiency of the photosynthesizing efficiency of such systems. Namely, the latest research, which are called in the literature the third generation of photosensibilizers, they concentrate on the modification of existing photosensibilizing dyes and polymers with external structures such as drug nanocarriers or antibody conjugates. And this is exactly the approach we have made in this work. Here is on this slide, you can see a Jablonski diagram showing the mechanism of photodynamic therapy. In short, after photosensibilizing dye absorbs visible light, it undergoes some internal changes, and after that, it can undergo two types of the photodynamic reactions, type 1 or type 2, which results in the generation of so-called reactive oxygen species that are they are toxic for the cells and well, they're toxic for the cells and they kill them usually through the apoptosis method, from which the treatment from which the therapeutic effect of the photodynamic therapy grows. On this slide, I would like to show you I would like to briefly show you some of the photosensibilizers that are already approved for the clinical use, such as the thermopropane, verteporfin, motaxafine lutetium, and talaporfin, and their respective trademarked names. Namely, the first presented photosynthesizer, thermopropane, is used by us in this work. Okay, now we would, and finally, to finish the slides, we have on this slide we have our the schematic representation of our studied three-component nanohybrids and its preparation stage. Firstly, we have first we have prepared the star-like copolymer with a dextran core and polyacrylamide arms via the radical polymerization process. After later, that polymer was converted into the highly anionic form. And finally, such a molecular system was used as a matrix for as a matrix for growing of the aurum nanoparticles inside in situ. And finally, to the resulting nanosystem consisting of the star-like dextran graft polyacrylamide polymer with aurum nanoparticles, we have added the thermoporphin solution and finally got our three-component hybrid nanosystem. We have studied the system and its individual components using dynamic light scattering method, and the resulting particle size distribution is present on this slide. I would like to note a bit peculiar particle size distributions for the dextran graft polyacrylamide aurum nanoparticles and the pure dextran graft polyacrylamide. Unlike the uncharged polymer solutions and and a lot of other things, which have a single peak distribution, these distributions, they have a complex multimodal structure, which we believe stems from the polyelectrolyte effects. After also there is data for the thermoporphin size distribution in the water I mean the pure thermoporphin in the water without any polymers. Thermoporphin in the water clearly forms aggregates, and we have two big peaks with two big aggregate peaks, one with the radius of the 145 nanometers and other with the radius of the about 10 microns, which is kind of expected because that pure thermoporphin is it's very badly soluble in water. For the more complex systems, such as dextran graft polyacrylamide with our nanoparticles thermoporphin and just dextran graft polyacrylamide with thermoporphin, we see the complete disappearance of multimodal structure, and instead they look and instead they resemble the characteristic distribution curve of the uncharged polymers. We believe that this effect is a result of the polyelectrolyte thermoporphin interaction, and we do not think that Gordon nanoparticles play any significant role in this process. So to tell you the exact numbers, the obtained radius for the thermoporphin polymer solution is 110 nanometers, and for the thermoporphin polymer our nanoparticle solution, the radius is 90 nanometers. Also, we have studied the absorption and the photoluminescence of our systems, which are presented on this slide. The absorption spectra of thermoporphin in water has features typical for thermoporphin, specifically the weak long wavelengths band Q peaks and intense short peak B peak. Such the characteristic features of the absorption spectra of bare thermoporphin is also present in in all of our prepared systems, namely in namely in the thermoporphin polymer and thermoporphin polymer golden nanoparticles, but are much more intense. Where the absorption magnitude is much higher. We believe that such increase in absorption intensity is based on the fact that after addition of the polymer, addition of the polymer breaks thermoporphin conjugates and increase its absorption intensity. And the addition of the golden nanoparticles to the mix produce us with a plasmonic enhancement effect for the absorption. So we believe that our absorption data proves both the plasmonic enhancement effect of thermoporphin absorption, which would increase its phototherapeutic efficiency, and also proves that our thermoporphin indeed binds with our study system. Also, these photoluminescent spectra have photoluminescent spectra also have the structure typical for the porphyrins in the work, typical for the porphyrins in our solute, but there is some different behavior after we add the golden particles. Well, similarly to the absorption factor, addition of the polymer increases the overall intensity. We once again believe that this is because thermoporphin conjugates disappear and gets incorporated into the polymer. But as you can see here, the addition of the golden nanoparticles indeed decrease the efficiency of photoluminescence. That's not as bad as it may seem, because actually the photoluminescence isn't what we are. Well, our idea isn't the increase of the photoluminescence intensity, but the increase of the reactive species oxygen and the general therapeutic effect of such a system. So we believe that this is indeed a positive effect, because it proves that there exists some kind of interaction between thermoporphin and golden nanoparticles, which is present in the photoluminescent spectra, which is once again proves the fact that our thermoporphin binds with the polymer golden nanoparticles nanosystem. Also, we have studied the time dependence of the photoluminescence intensity, and there is some interesting results. While there is no significant change in the photoluminescence intensity of bare thermoporphin in water, the temporal behavior of the photoluminescence of thermoporphin in the composition of thermoporphin dextran polychloramide and thermoporphin dextran polychloramide gold nanoparticles is fundamentally different. Specifically, during the first 30 to 60 minutes, the intensity sharply increases, after which it remains constant. We believe that this is the result of ongoing binding process of thermoporphin with our polymer nanosystems. Whereas after also it's not present here, but in the 2 or 3 days after the mixing, photoluminescence intensity starts to drop. Okay, and finally, there is also finally, there's also the results of our concentration research on such systems. As you can see, the increase of thermoporphin concentration, well, naturally, it leads to increase in the absorption and the photoluminescence intensity. But there is some interesting effect of the increase of the polymer and Gordon nanoparticles concentration. In general, at minimal concentration of the, for example, after some level of gold and polymer concentration, we see that we see the seen before effect that indeed the intensity of photoluminescence decrease after the intensity of photoluminescence of thermoporphin polymer gold solution is lower than intensity of photoluminescence of thermoporphin polymer solution. We believe that this is a result of non-radiative energy transfers from the thermoporphin molecules to the golden nanoparticles and just a general damping effect. Namely fat for them. Also, to finally reach the more biological part of our research, we have studied the generation of the reactive oxygen species under photodynamic therapy conditions for our nanosystems. No statistically significant difference in the reactive oxygen species levels were found after irradiation with 600 and 6 nanometer lights for water and for solutions of water and dextran graft polyacrylamide with golden nanoparticles. However, there is a significant reactive oxygen species level in the pure thermoporphin in water, and there is even bigger increase, almost double the increase compared to the bare thermoporphin in water. In our study nanosystem, polyacrylamide, golden nanoparticles, thermoporphin. So we have indeed proved that such nanosystem generates reactive oxygen species much more effectively compared to the bare thermoporphin in water, which would be useful for the photodynamic therapy applications. Also, there is the other biological research. The potency of the nanocomposite was tested we tested the efficiency of the nanocomposite in the triple negative breast cancer cells and human epithelial disc cells. As you can see here, the cell counts in the triple negative breast cancer on the right with the red graph have decreased significantly after photodynamic therapy, well, after the light irradiation using our triple hybrid nanosystem. Something that should be noted that decrease is even higher compared to the normal epithelial breast cancer, which proves that our system is indeed can be considered targeted system, effective effect, which would affect cancer cells considerably more than normal cells. And also we have studied the effect of individual components of our system for on the triple negative breast cancer and the human epithelial cells. The most important here is the fact that after light irradiation of bare thermoporphin, there was still around the 40% of the triple negative breast cancer left, but after the irradiation of but after the photodynamic therapy, the light irradiation, of using our system, there was just 20% triple negative breast cancer cells left. So to jump to the conclusions, we have observed almost a twofold increase in reactive oxygen species generation by our prepared three-component dextran graft polyacrylamide, our golden nanoparticles, thermopolymorphin nanosystem under 660 nanometers LED light excitation compared to the water solution of bare thermopolymorphin. And also we have demonstrated that our studied three-component nanosystem is applicable and effective for an in vitro treatment of triple negative breast cancer. And that would be all. Thank you for your attention.
Okay, thank you, Pablo, for your report. Does anybody have a question? We have one question from the audience. Two questions.
Please explain the mechanism by which gold nanoparticles have an effect on the bioactivity of a photosensitizer, thermoporphyrin. How can they enhance the efficiency of photosensitizer?
Yes. First, we believe that due to the plasmonic enhancement of the light absorption by photosensibilizers, they absorb light more efficiently, so they generate reactive oxygen species more efficiently. And also because the loss generation process is in itself the photodynamic process, it is also increased by the plasmonic enhancements. Well, basically, the plasmonic particles generate a strong localized field around themselves, which enhance all the photo processes in their vicinity, like absorption, because it's a photo process, obviously, and the absorption is something that we can definitely see that was increased, but also we believe that the generation of reactive oxygen species itself got enhanced by such intensive electromagnetic field. And that would be my answer if it's fine.
Some again question, why have you choose exactly gold nanoparticles, but not, for example, silver nanoparticles, as you have seen the most enhanced B band, about 400 nanometers, and the maximum of gold nanoparticles is about 530 nanometers. Also the question to show the nanoparticles activity, I expect you will show some triplet luminescence, but not singlet luminescence. And in your biological testing, you have not show the result for the system with the polymer and this porphyrin dye, but within nanoparticles. While in absorption and luminescence, this system with dextrin and this porphyrin dye show very similar results to the same system with addition of nanoparticles. Thank you.
Yes, about the silver nanoparticles, we just haven't. We are working on this. It's the working process right now. We have only the results with the gold nanoparticles. And why this? Yes, biological exchange. The results of the polyacrylamide with thermoporphyrin without the gold nanoparticles isn't present here, but it is practically the same as well, okay. It's they show practically the same results as just a thermoporphyrin system. Well, I mean thermoporphyrin with polymer without gold nanoparticles in the well in the cells in the experiment with the cells. Have I missed something, or would that be all?
Okay, maybe one more question. Oh, thank you, Paolo, for your report. Thank you all for discussion. And we move forward. I invite for the report PhD student Yulia Kulish with topic the effect of single-walled carbon nanotubes and nanographene oxide on expression levels of EOB1, PKGS2, and HSPA5 in normal human astrocytes.
Good afternoon for all who are on site and online. It's like I wanted. I have some. Okay, so I can begin. Yes. Do you hear me well?
Please launch in full screen mode your presentation.
1 second. Do you see it now? No? Is it on full screen?
No.
No? 1 second. If I share in, I should share whiteboard or presentation? Or what? Presentation, okay.
Try to launch full screen mode and then share your presentation.
Okay, sorry. Sorry about this. Okay, so now
not okay.
Oh, I don't know why it. So maybe now? Because I see it on full screen, but you not, yes?
Yes.
Okay, I can begin like
just
in this view, maybe.
You need to start your presentation, and after this, in Zoom, share screen, your full screen mode presentation.
I'm sorry.
And now start your presentation.
Mm-hmm.
And then in Zoom,
Mm-hmm.
share screen your presentation in full screen mode. There must be an icon.
Yes, I see it. Oh, 1 second, 1 second. If I can do it. No. Oh. Так. Maybe now?
Okay.
No?
Okay.
It's okay. It's okay.
Okay, I can begin. Can I begin? Oh, okay. I don't know. Okay, so
Please take your flow.
yes, thank you. My the effect of single-walled carbon nanotubes and nanographene oxide on expression level of TOB1, PTGC2, and HCP5 in normal human astrocyte. Our work was at Department of Molecular Biology of Paladin Institute of Biochemistry of the National Academy of Science of Ukraine. So, you can see. You cannot see how I listed slides, yes?
Yes, we doesn't see your.
I'm very sorry about this. Oh, now can you see it?
Yeah.
Yeah. So the study of molecular mechanism of action of nanoparticles, including carbon nanoparticles, on genome function, expression of key regulatory genes, including transcription factors, and in normal human astrocyte, is extremely important because it allows to identify today possible negative consequence of the use of nanoparticles in biology and medicine. For example, single-walled carbon nanotubes are often used as transporters for drugs in treatment of cancer, like with doxorubicin and other drugs. The aim of study was to investigate. The aim of study is was investigate the sensitivity of the expression of TOB1 and some other genes associated with endoplasmic reticulum stress and cell proliferation to the low doses of single-walled carbon nanotubes and nanographene oxide in normal human astrocytes. Role of genes. That's why is demonstrated on slide, and that's why we are choose them, because TOB1 is one of important anti-proliferative protein, and it's important in proliferation of cells and for normal cells and for cancer cells. So PTGS2 is important because this synthase is involved in biosynthesis of metabolism of fatty acids, lipids, prostaglandins, and its cancer-related gene. And chaperone or heat shock protein family member 5, it's important in this chaperone is important in stress endoplasmic reticulum stress because it's like one of some of important transcriptional factor that begins some signal waves. So on the slide, it showed on the slide materials and method. We used normal human astrocytes. It's line NHTC. Is NHTC. Oh. And we used single-walled carbon nanotubes in concentration 2 and 8 nanogram per milliliter in medium, and nanographene oxide in concentration 1 and 4 nanogram per milliliter for 24 hours. So single-walled carbon nanotubes was 1 and 2 nanometer in diameter and were suspended in distilled water and filtered through membrane filter millipore size 1 micrometer and No, it's not 1 micrometer. We used we broke up with ultrasound and filtered through pores with 0 to micrometers. So these carbon nanotubes were received from Professor Uwe Ritter and later with Professor Yuri Prelutsky for our department, and my chief, Professor Alexander Minchenko, worked with them. And I worked with RNA and these nanotubes in our department. So on the slide on the slide demonstrate some information about these nanotubes and about results. So the conducted studies revealed that in the culture of immortalized normal human astrocytes and with medium with single-walled carbon nanotubes and nanographene oxides, we see some effects of expression level of TOB1 genes, PTG, C2, and HCP5 mRNAs, which are associated with endoplasmic reticular stress and cell proliferation. On this slide, you can see the impact of single-walled carbon nanotubes that was in medium in concentration 2 and 8 nanogram per milliliter for each gene. And it significantly increased expression level, significantly increased for HCP5 and significantly decreased for TOB1. So and that's a result for nanographene oxide. In these results, we can see that level of expression TOB1 was increased and level of another genes were decreased. Compares in TOB1 level, but it still increased compared to control. So it's quite possible that this change in genes expression are mediated by endoplasmic reticulum stress due to the action of carbon nanoparticles, and therefore the low doses of graphene oxide and carbon nanotubes can disrupt the functional integrity of the genome and possibly have a genotoxic effect. So it's required for careful assessment of potential exposure-related risk for human health. That these data suggest that more caution is needed in biological and medical application of single-walled carbon nanotubes. Thank you for your attention, and glory to Ukrainians and glory to heroes.
Thank you for your report. Does anybody have some question? We have no questions. Thank you once again.
Thank you for your patience with my problems of sound.
That's okay. So the next speaker, PhD student Maria Khutko. She report about seed silver nanoparticles coating with thermoresponsive polymer PNIPAM synthesis and characterization from Lashkarov Institute of Semiconductor Physics of the National Academy of Sciences of Ukraine. Please take your floor.
Good afternoon, dear colleagues. Today I represent you a little work about silver nanoparticles coating with thermoresponsive polymer penny palm, synthesis and characterization. If I want to talk in general, the global target of our work is investigation for creation of drug delivery systems with thermoresponsive polymers. And maybe further aim is to create a sensor based on fluorescence and with a polymer like a spacer to manipulate the fluorescence. So in the first stages, we encountered a problem that the citric coated nanoparticles somehow aggregates. And we tried to solve this problem. First of all, we wanted to create a polymer layer instead of citrate, but this replacement crashed. Then we used inverter. Recently, we had a good experience with molecular implanted polymers, and we took inverter from the synthesis of that polymer. So here you can see the reaction. Here's our inverter, and it has two sulfur groups that serves for controllable radical polymerization, and also it has a thiol group. It serves for good binding with noble metals. Left is an isopropyl acrylamide. That is a thermoresponsive polymer with low critical temperature, 32 degrees, and it's our resulting polymer. It's a typical spectra of clear penny palm and isopropyl and inverter isopropanol. You may see that they absorb light near 440 nanometers. First of all, we took silver nanoparticles coated by citrate and synthesized by standard protocol. They absorb light on 400 nanometers and added that inverter to detect while it's aggregate. And as you may see, the blue line that our peak disappears. Also, the color of the solution became from yellow to colorless. Then we decided that we want to make our system and polymerization simultaneous with the creation of nanoparticles. So we mixed inverter, penny palm, monomer NIPAM, and argentum nitrate. And as you may see, there are two peaks. One that indicates the creation of nanoparticles, and the addition of borohydrate didn't change this situation. Also, there is really light scattering because there are many polymers, free chains. in the solution, but during the process of synthesis, this relay scattering decreases because these free polymer chains bind with our nanoparticles. Then we try to investigate the stability of our nanoparticles. And as you may see, while we took the concentration in 10 times larger than in previous experiment, even after 10 days, it's still stable compared with other one. Also, we used ultrasonic bath because some nanoparticles settles on the walls of the vessel. On this slide, you may see that our nanoparticles are really yellowish compared with clear solution of argentum nitrate, and seed-cell nanoparticles are synthesized by a standard protocol. They have the same concentrations. On the left spectra, it's the same comparison, just on one spectra. Also, from this spectra, you may see that the formation of polymer is much quicker than the formation of nanoparticles. So in further investigations, we will try to increase the yield of our nanoparticles. If we talk about the sizes of these silver nanoparticles, they have interesting result because it's somehow too big. We explained that because of the halide complexes that form and polymer and monomers with argentum ions and argentum nanoparticles. We confirmed this halide complexes by calculations in program PC model. We took the lowest energy of this. I will show you in the next slides. Okay. And also we tried to calculate stability of these complexes also in this program by dynamic methods. It's formation of our complex from left to right and then lower. And also stability. As you may see, just one molecule of monomer NIPAM comes out of the complex. So the conclusions. Polymer-coated noble metal particles can be used in various fields, such as drug delivery and sensors. Using a specialized inverter with the old group for binding with the metal makes raft polymerization much easier and with fewer stages. The creation of a thermoresponsive polymer layer simultaneously with the synthesis of seed silver nanoparticles was successful. UV visible absorbance spectra prove the stability of silver nanoparticles with the increasing of NIPAM and in further concentrations. The formation of the polymer is performed much quicker than the formation of the silver nanoparticles. NIPAM or PNIPAM form electrostatic halide complexes with argentum ions and nanoparticles. Also, this work was supported by a National Research Foundation of Ukraine. Thanks.
Thank you for your presentation. Any questions, please? We have one question. Just a moment.
Thank you for your presentation. What part of PNI-PAM polymer is responsible for thermo responsive effects, or what mechanism is lie behind it?
Thank you for your question. May you return the presentation, please? In general, for thermoresponsives, it's very important to have a balance of the groups. One moment. It's important to have a balance of the groups that are water soluble and non-soluble. Here we have a non-soluble group with a quite long chain, and more soluble is with secondary, thirdly amine. In solution, there is how to explain? When the temperature is lower than a low critical solution point, it has a random situation in the solution. But when it's lower, the entropy of our system grows, and the propylacrylamide becomes more like cell or global. Maybe I answered your question.
Okay, which is approximate response time of this polymer? What time takes it to free what's inside it?
We didn't measure the response, but I may say that very heated solution, very how to say, it's more not colorless, but it's visible. You may see that it changes its state.
Okay, thank you very much.
Thank you. The last question.
Okay, then I ask stupid question. Maybe why do you have why the extinction goes up for the wavelengths less than 400 nanometers in your graphs? Like here or like here.
Yeah.
Yeah. So we see that at your graphs, the extinction goes very high, right? For the wavelengths less than 400 nanometers.
As I said before, it's because of the relay scattering, because there are many polymer chains in the solution, and only half of these polymer chains may bind with a nanoparticle on this stage, because it's a special mechanism when monomer goes to the one sulfur that is dormant, and its group goes out, and then there is a polymerization goes. And the second stage is when a radical goes to this sulfur that is like with two bonds. Our long polymer chain also goes out, and then the polymerization goes with the sulfur. So you have two situations when our polymer binds with an inverter and then with another particle, and other situation when it's free in a solution.
Okay, thank you.
Thank you for your questions and your report. And the next speaker, PhD student Oksana Kolesnikova from Bogomolets Institute of Physiology of the National Academy of Science of Ukraine.
Good day to everyone. Thank you for the opportunity to take part at the conference. Let me introduce my report. Gold nanoparticles have a size-dependent effect on spermatogenesis in mice. Okay, and you know that today is very important to study and investigate gold nanoparticles, functional gold nanoparticles, and now we have some investigation in this field, for example, in theranostic, in targeted drug delivery, and photochemic treatment, and another. But the literature data on the ability of gold nanoparticles to penetrate the hematotesticular barrier and causes some impairment or some treatment effect of the morphofunctional state of the organs of the male reproductive system are still controversial. Earlier, we studied gold nanoparticles with polymer matrices. It was star-like polymer matrices with a graft core and polyacrylamide chain. A few report earlier, we listened this report in photodynamic therapy, for example, and we studied these gold nanoparticles in this system under normal condition and in experimental pathologies. It was like diabetes and chronic kidney disease. And we found that these treatments have some potential therapeutic application for improving male reproductive system. But in these gold nanoparticles in these polymer matrices have different sizes. It was from 4 nanometers to 15 nanometers. And in our investigation, we want to investigate standardized size of gold nanoparticles. And we want to evaluate the effect of 5 nanometers and 10 nanometers gold nanoparticles from sigma on spermatogenesis in mice under condition of experimental hyperglycemia. And it is about our gold nanoparticles. It was from sigma. We bought these nanoparticles from sigma. It was 5 nanometers, one bottle with 5 nanometers and another with 10 nanometers. And these gold nanoparticles were spherical in shape. It is similar to gold nanoparticles in our previous studies, also spherical shape. And we injected these gold nanoparticles intratesticular once a day, 5 times. And our experimental model was experimental model of hyperglycemia. And we administrated, it is a classical model of hyperglycemia. It is also named diabetes. It was 120 milligram per kilogram nicotinamide. And we wait 15 minutes. And after this, we injected 70 milligram per kilogram streptozotocin. And we do this in first, third, and 5th day of the experiments. In this picture, you can see more detail our experiments. It was hyperglycemia first, fifth, and third and fifth day of the experiments. It was in vivo, and we wait 1 week because during these protocols we need 2 weeks for waiting to create these models, and after this we injected intratesticular 5 nanometers of gold nanoparticles and 10 nanometers of gold nanoparticles. And in the end of these experiments, we have 2 series. In one series, we investigated germ cells, spermatogenesis. In the second, we investigate fertility. It was pre- and post-implantation mortality and number of live newborns per mile, per female, sorry. And it is our results. First, it was a ratio of cells of different generation of spermatogenesis epithelium. We investigate spermatogonia, spermatocytes, spermatids, and cells of Sertoli cells. And upper part of this table, it is about 5 nanometers of gold, and level part, it is about 10 nanometers of gold. And we found that under condition of hyperglycemia and injection of 5 nanometers of gold nanoparticles, we have increase of number of spermatids and increased number of spermatocytes, which indicates improvement of male reproductive system, improvement of spermatogenesis. But when we injected 10 nanometers, we did not find different changes between these 2 groups, between group of hyperglycemia and gold nanoparticles. Okay, and next we study spermatocytes, and we investigated number of living apoptotic and necrotic cells of the tested, and also we found that number of necrotic and apoptotic cells decreased, and number of living cells is increased, and it is also indicate about improving of spermatogenesis. But under condition of injection of 10 nanometers of gold nanoparticles, we did not found changes between this group, between group of hyperglycemia and hyperglycemia plus gold nanoparticles. Okay, and next parameter was number of sperm, a concentration of sperm, millions per milliliter, and number of abnormal form of sperm. We study total number of abnormal sperm and primary anomalies. And under condition of hyperglycemia and injection, as 5 nanometers of gold nanoparticle, as 10 nanometers of gold nanoparticles, we did not found any change under these conditions. And next was investigated number of living apoptotic and necrotic cells of the epididymis. It was spermatozoa. And also under this condition, under condition of hyperglycemia and injection of gold nanoparticles, we did not found anything change in these parameters, and it can be explained by spermatozoa need more longer time for a complete cycle of spermatogenesis. And the last, our part on the investigation, it was pre- and post-implantation this rate and number of live newborns. Also under this condition, like gold nanoparticles in size 5 nanometers and 10 nanometers, we didn't see anything change between this parameter, between hyperglycemia and hyperglycemia plus nanogold. This data connect with data about spermatozoa, because we didn't have some changes in spermatozoa. That's why we did not have any change in fertility. Okay, and last one about conclusions. So, under condition of experimental hyperlipemia, intratesticular administration of 5 nanometers gold nanoparticles from sigma improves spermatogenesis in mice, but administration of 10 nanometers gold nanoparticles from sigma does not lead to change in spermatogenesis. Gold nanoparticles have a size-dependent effect on spermatogenesis in mice. This particle size is the key, but not the only parameter that determines the biological action of both of the particles. Thank you for attention.
Thank you for your presentation. Question, please.
Thank you. Without question.
Your question.
All right. Okay, thank you for your question. It's interesting because, as you see, in all groups, we have some control in gold nanoparticles. Yeah? And we did not have anything change in this parameter because it is a small concentration of gold nanoparticles, and its low concentration in normal position did not change this parameter. And it is and under some pathology condition, these gold nanoparticles can lead to some changes as toxic as a treatment effect. I think it can be connect and compare some reasons for this. I think it's like various reported about these gold nanoparticles. This can change some balance of reactive species inside in these cells. I think it's reasons for this. Thank you.
Thank you for your question and thank you for your report. Now we go into the next session of our Nanoplasmonic and surface and spectroscopy. The chairman of the section is Dr. Olena Hasenko.
Good day, everyone. And now we are moving to our session. Nanoplasmonics and surface enhanced spectroscopy. We have only 3 slots. Our first speaker, Oleh Dimitri, he is from Kyiv. Last period Institute Semiconductor Physics of National Academy of Sciences, Ukraine. Dear Lord, Dimitri, are you with us?
Yes, I'm here.
Please,
Can you hear me?
Yes, yes, everything is okay. You can share.
Okay. Can you see my screen?
Yes. Very
Yeah, okay.
funny picture at the first.
Yeah, good afternoon. And today I would like to tell about application of plasmon nanoparticles for enhancement of near-infrared upconversion luminescence. First of all, what does it mean, upconversion for photoluminescence? This is the process where the emitted energy of the emitted photon is larger than the energy of the absorbed photon. And for this process, in order to follow the energy conservation, we need the participation of some Maxwell daemon who support this process. And I will show you who will play the role of this daemon in my presentation in the next slides. So why upconversion photoluminescence is important? This process can be applied for different processes, for example, for upconversion of thermal energy, for near-infrared vision, for harvesting near-infrared energy by solar cells. Because we know that solar provides us energy which is spread in the wide spectral range from ultraviolet to infrared, and infrared domain provide about half of the total energy which come from the sun. But this domain is not properly used. For example, for operation of solar cells, because most solar cells harvest energy in the visible range. Some solar cells harvest energy just in the beginning of the near infrared, but very few can operate in the middle or far infrared. So if we are able to convert this energy from the infrared range to the visible range, then we can provide more energy for operation of solar cells and to provide higher efficiency of these devices, respectively. So upconversion photoluminescence can be achieved by multi-photon processes and single photon processes. I will tell only about single photon process. And here you can see the comparison of the usual Stokes photoluminescence when we excite electron to the first excited level, from which the exciton relaxes to the ground state and emit light. And energy of this light does not exceed the energy of the absorbed photon. And the upconversion photoluminescence, in this process, we need first to lift the electron from the ground state to the hot band, which consists of the continuous of energies of vibronic levels. And for this process, we need some heat. And this how the Maxwell demon should work. It should lift the electron to some vibrational level, and then this electron can pick up the photon of a lower energy than the photon which then can be emitted, because in the emission process, the photon relaxes from the first excited state to the ground state. But technically, the energy of this emission is the same as the energy of the emission in the usual Stokes process. And here you can see the comparison of this of the spectral features. We can see the position of the emission band is the same, but the excitation is different. In the Stokes emission, we should excite this process with higher energy, but in the anti-Stokes emission, we excite the system with the lower energy than the emission wavelengths. A hot band, which I told about, can be identified as a small bump in the absorption spectra. You can see here at longer wavelengths compared to the absorption of the main band gap absorption. And the special feature of this hot band absorption is that it increases with temperature. This opposite to the main absorption band, which decreases with increasing temperature. So here you can see this process when we increase the temperature from room to 80 degrees, the hot absorption increases. And it increases linearly, approximately linearly with temperature, both when we increase temperature or decrease temperature. Similarly, the anti-Stokes emission, anti-Stokes photoluminescence also depends on the temperature, and it increases when we increase the temperature. And this process also approximately linear. The anti-Stokes photoluminescence intensity increases linearly with temperature, both when we increase temperature or when we decrease temperature. So temperature can play the role of enhancement of the anti-Stokes emission. But certainly this option is not optimal, because for this process we need the energy consumption by the system. So we will concentrate on the other process, how to increase this anti-Stokes photoluminescence by plasmonic nanoparticles. It should be mentioned that anti-Stokes photoluminescence give a relatively low quantum yield. So in this table, you can see the comparison for 4 infrared dyes, which have Stokes emission. In this column, you can see the quantum yield of the Stokes emission and quantum yield of the anti-Stokes emission, which is much lower compared to these values. Therefore, in order to make the process of energy upconversion efficient, we need to enhance anti-Stokes emission by some process. And this process in this work is the application of plasmonic nanoparticles. So specific of anti-Stokes photoluminescence requires requires attention of 2 points. The first point is the population of the hot band, because without this population, we do not have efficient hot band absorption and anti-Stokes photoluminescence respectively. So plasmonic nanoparticles should play the twofold role. First, they need to provide heat to the system in order to increase population of the hot band. And the second role is to enhance photoluminescence from the first excited state to the ground state. And we know that, for example, gold nanoparticles can produce these can play these 2 roles, because when we excite a plasmon in gold nanoparticles, gold nanoparticles simultaneously increase the temperature if we apply a relatively high excitation energy, then these nanoparticles increase the temperature as well. But we cannot apply a usual spheric nanoparticles, spheric gold nanoparticles for this process, because we know that spheric gold nanoparticles provide plasmon resonance only in the visible range, near 540 nanometers. But for near infrared region, we need the other particles, which have plasmonic resonance just in the near infrared. And for these purposes, we selected 4 different nanoparticles, but 3 nanoparticles have the same core, which is the copper sulfide core, and they have also the silicon oxide shell of the different thickness from 11 to 25 nanometers. And this shell plays the role of the spacer between the plasmonic nanoparticle and the dye, which is absorbed on the surface of this core shell particle, because we know that the direct contact between a nanoparticle and the emitter results in quenching of photoluminescence, which is unwanted process. So we need some separation between the surface of plasmonic nanoparticle and the emitter. And the second type of nanoparticle was gold nanorods, also covered with the silicon oxide shell. And these nanorods have two plasmonic resonances, longitudinal and here you can see on this absorption spectra two resonances. The first one is near 40 nanometers, which is a usual resonance for gold nanoparticles, and the second, due to the longitudinal plasmon excitation, is located in the near infrared at about 1000 nanometers. And copper sulfide nanoparticles have plasmonic resonance near 1200 nanometers. So we applied these nanoparticles in order to study the effect of enhancement of anti-Stokes photoluminescence. For these purposes, we used two different dyes, which are shown here, and which are different by the structure of the mesogroup here, near the central part of the chromophore. So, the purpose of this difference was to study the interaction of these molecules with silica surface of the nanoparticles, either by sulfonic groups or by the amino group at mesoposition. So here you can see the absorption, photoluminescence excitation and photoluminescence emission spectra of these dyes, which are located in the near infrared. And also here you can see the absorption spectra when the nanoparticles are mixed with these dyes. And specifically when the first dye did not show any evidence of good interaction with gold nanoparticles or with copper sulfide nanoparticles, because the spectrum, you can see this spectrum is practically the same when we have dye absorption shown by black line and dye in the presence of nanoparticles shown by blue line. But for the second dye, which has amino group in the mesoposition, we can observe a clear interaction because the spectra become changed. Especially for this dye, we have two absorption bands. The first band is located in the visible, and the second one locates in the near infrared. And this is due to the different conjugation of the molecule when a mesogroup interact with the silica surface. For example, if we have a silica surface which are more acidic, then we have increase in the conjugation length of the molecule and increase of this infrared band and decrease of the visible band. But if we have basic character of the surface, then we have deprotonation of this amino group and the change of the conjugation lengths, which become smaller and which produces the increase in the visible absorption band and decrease in the near infrared band. So we can conclude how this dye interact with the silica surface of nanoparticles. And further, we study the photoluminescence spectra in the presence of these nanoparticles. So here you can see these two dyes in the presence of gold nanoparticles. And specifically for the first dye, the spectra of the dye excited at different wavelengths, whose energy is smaller than the emission energy, of course, because here you can see the anti-Stokes photoluminescence. And compared with the same spectra in the presence of gold nanoparticles are practically the same. And the intensity of the photoluminescence as a function of the excitation wavelengths, you can see here for the dye in the absence and dye with the presence of gold nanoparticles is approximately the same. There is only small difference at specific wavelengths. And if we have the ratio of the intensity of photoluminescence of the dye with nanoparticles and without nanoparticles, then we can see this ratio shown here for dye 1 by black symbols, which is less than 1, less than unity. That means there is no enhancement of anti-Stokes photoluminescence for this dye. But for the second dye with amino group in the meso position, we have small increase of photoluminescence and this increase depends on the excitation wavelengths. When we change excitation wavelengths closer to the plasmonic resonance wavelengths, then we can observe a higher intensity. That means the better enhancement of the anti-Stokes photoluminescence. And you can see here on the bottom figure, the green line shows that the ratio of anti-Stokes photoluminescence in the presence of gold nanoparticles to the photoluminescence of dye without nanoparticles increases and become larger than unity. That means we have true enhancement of photoluminescence here, and this enhancement increases when we approach excitation wavelengths to the excitation to the plasmonic resonance shown here by vertical line. Then let me tell about interaction of these dyes with copper sulfide plasmonic nanoparticles. And here you can see the three-dimensional excitation emission spectra, which can tell us something about how the spectra change when we add copper sulfide nanoparticles. And for the first dye, you can see that the spectra are practically three-dimensional map practically do not change when we use two different copper sulfide nanoparticles with different silica thickness. But for the first dye, we can see clear change, especially for the bottom figure. We can see the extension of this emission features compared to the features of the dye without nanoparticles. And specifically, if we look at dependence of photoluminescence intensity at the function of the wavelengths, we can see that for the second dye, dye number 2, we have here you can see blue curve, which shows deflection from the black one. Black one corresponds to the anti-Stokes emission of dye without nanoparticles, and green in the presence of nanoparticles with the specific silica thickness. We can see clear enhancement. And the ratio of the intensity of anti-Stokes emission in the presence of nanoparticles and in the absence of the nanoparticles is shown here. Again, we can see a clear enhancement at specific wavelengths, which are close to the wavelengths of the plasmonic resonance. And this enhancement reach up to 50% for this case, for this dye and for this specific copper sulfide nanoparticle. So I summarize these results. We achieved enhancement of the anti-Stokes emission for specific dye, which has amino group in the meso position. And this enhancement, of course, is due to the specific interaction of this dye with silica surface, because this dye just interact by amino group with silica surface, and this provides just better conditions for the enhancement of the photoluminescence. And we reached about 17% of enhancement when this dye interact with gold nanorods, and about 50% of enhancement when this dye interact with copper sulfide nanoparticles. So my conclusions is that we found specific dyes which have anti-Stokes photoluminescence, where the anti-Stokes shift can reach up to 100 nanometers, and where quantum yield of this anti-Stokes photoluminescence can reach 10% when we excite this dye with specific wavelengths. Then we found that this specific dye can reach enhancement of anti-Stokes photoluminescence when interacting with gold nanoparticles by 17%, and when interacting with copper sulfide nanoparticles by 50%. I would like to acknowledge people who contributed to this project, who helped with materials and studies. I would like to thanks Japan Society for Promotion of Science for providing the grant for the specific research work, and thank you for your attention.
Thank you for your presentation, and now we have time for question. As I understood, online we didn't have question, maybe in the hall. Yes, in the hall we have one question, and after this I also have one question for you. One moment, you didn't turn on a microphone and he cannot to hear you.
May I ask you whether you collaborated with some chemists from the Institute of Organic Chemistry of the National Academy of Ukraine? I ask this because we have such dyes in abundance. And as far as I understand, the structure of dyes is of primary importance in your case. They should be near infrared. They should have some specific structural features. So it is simply interesting for me whether you have some contacts with my colleagues. I mean, those who synthesize cyanide dyes with bridge structures in the meso position in the Institute of Organic Chemistry.
Yes, sure. This one of these dyes, which I showed you, was synthesized by chemists by Dr. Slominsky in Institute of Organic Chemistry, and the second dye was synthesized by Japanese chemists. And of course, I collaborate with Institute of Organic Chemistry. Unfortunately, people passed away. Recently, maybe you know, Professor Ishenko passed away, and I collaborated with him, and we had some agreement on research work in this direction. But of course, I want to continue this collaboration. And unfortunately, I cannot see your name in this.
Yes. Can you tell your name?
Dr. Marina Dekhtar, just from the department headed by Professor Academician Ishenko that passed away a fortnight ago. Yeah.
Of course. And I lost this contact, and I want to to find contact of other people in order to continue this work. If you can provide your contact, we will collaborate, sure.
I think it is possible to put even in the chat. You can put.
Yeah, yeah, I would appreciate if you leave this contact for me.
Okay, it's a nice example when during the conference start new cooperation. And now a question from my side. As we have opportunity to listen your report, your research is more focused on enhancement of photoluminescence. But the question, if you try to use this dye for surface enhanced Raman spectroscopy, or maybe a surface enhancement infrared spectroscopy, yes, I realize that it is dye more for photoluminescence, but sometimes it's possible to see enhancement in different methods.
No, I did not try these dyes in surface enhanced Raman spectroscopy. Yeah, but yeah, we can collaborate in this direction as well. If you have any idea, so we can try.
It's possible to modify. Okay, thank you a lot for your report. And now we are moving to our next speaker. I prefer to check if Professor Leonid Fedorenko with us.
I'm here. Thank you.
Nice. Please take your floor.
Share. And share. Sure. Do it. It's on.
You can see your slides, but not in the full screen mode. But if you prefer, you can continue in such way. But I'm afraid a little bit that maybe your slide in the future will be not moved because it is not full screen mode.
You see my report?
Yes, yes, we see your report, but it is not full screen mode.
It is necessary. It is convenient for me in such form, if you agree.
You can,
okay?
You can try, but as I know, sometimes it's happen if it is not full screen mode, your slides cannot move. Can you please check if your slides will move? Can you press next slide? We will only to check.
Yes. I try. For example.
Okay, so everything is okay. So please start.
Thank you. Good afternoon, dear colleagues. My report relates to the laser technology of aluminum nanoparticles formation in conditions on surface plasmon resonance. Attention to aluminum plasmonics arrived after 205 Renaissance because of the first info about surface plasmons appeared namely for aluminum in 1957 up to 59. Since the 1990s, the efforts of the researchers have been focused on the aurum and argentum as metals with plasmonic resonance in the visible, more convenient and widely used part of spectrum. A significant difference of aluminum plasmonic lies in the possibility to excitation of bulk plasmons in the ultraviolet range, about 230 nanometers, due to higher number of valent electrons, equal 3 for aluminum and equal 1 for argentum for aurum and aurum. Next. Yes, sorry. Among the positive features of aluminum plasmonics, we were particularly interested in the ability of expand and shift the extinction band at plasmonic frequency into the long wave direction depending on the size of nanoparticles. Increase of the nanoparticle size leads to expansion of the extinction band and shift of the maximum position to the near infrared up to the 60 and more nanometer. This fact prompted us to study of the possibility of laser-assisted nanofragmentation of thin films, aluminum films, by irradiation of the visible spectral range due to forced plasmons excited under surface resonance conditions by high-power pulse of Young laser irradiation at 532 nanometers. Yes, theoretical evaluation. We consider surface electromagnetic waves in the form of standing plane waves with the field intensity in this form. The resultant sensor electromagnetic wave intensity is presented in the next form. We also considered the dimensionless thermal conduction equation with the following initial conditions. There are the dimensionless parameters. Sorry. Okay, so oh, this one. As a result, this solution gives the following graphical dependence. This is a thick line correspond to the laser intensity, and thin line correspond to the temperature distribution of the X-ray. And the horizontal dashed line indicates a maximum value of dimensionless temperature, which is the consistence of solid and phase and boiling aluminum would be possible. Aluminum would, yes. The distribution of the hot spots, dimensionless temperature on the metal film surface. So, next one. Yes, experiment. Two types of the structure were used in the setup arrangement. First type of structure, the glass with aluminum nanofilm, thin nanofilm, the so-called donor structure, and this second structure, this glass I2 layer and polymer layer, is acceptor structures. So small polyreturn waves were excited in the donor structure by excitation with powerful pulses of second harmonic of the Young laser with a wavelength 522 nanometers. In the Kretschmann configuration, these hot spots are formed on the surface of the metallic layer as a result of lateral temperature gradients in the aluminum thin film. Temperature gradients were stimulated by interference of plasmon polyreturn waves, then leads to appearance of the molten aluminum fragments and high enough laser intensity. When the laser intensity exceeds the microablation threshold, the aluminum fragments transfer through the air gap and implant the polymer layers in the second acceptor structure. So experiment. Yes, experiment is result of the experiment. Figure 4 shows the images of the structure surfaces. AB is donor. This is acceptor structure with aluminum nanoparticles. This is AB and NB. Its S picture shows the donor structure after laser microablation, and C and D image shows the same images with other particles in range of size from 15 to 300 nanometers. So, Raman spectra. Figure 5R shows the Raman shift spectra for the various formation stages. Final acceptor structure, aluminum polymer film I2 glass plate, magenta color, and donor structure, donor structure aluminum thin film, glass plate, black color. Let's consider Raman shift spectra in the final acceptor structure. This band maximum 1003. This is magenta. Yes, magenta color. The band maximum 167. 67 centimeter, inverse centimeter, gets a slight low frequency shift after aluminum nanoparticles implantation into the polymer film from 1,367 to 1,360 inverse centimeter. The band shift of 7 nanometers, there is a result rather of the aluminum nanoparticles appearance that is confirmed by the deconvolution figure B. You can see green color, this band. Sorry. Next. Reflection and transmission spectrum. Reflection spectrum of the final acceptor structure is shown in the green color, The aluminum nanoparticles implantation to the polymer layer causes some deepening at 250 nanometers and appearance of the shelf near the 280 nanometers, as compared with previous spectrum, black color with only polymer layer I2 and on glass plate. This shift can be explained by a resonant absorption of localized plasmon of aluminum nanoparticles implanted to the polymer layer.
I'm sorry, please.
Sorry, conclusions?
About your time, you have maximum 3 minutes, okay? No, you have 3 minutes, but not more, okay?
You can see. There is conclusions. There are conclusions. The possibility of a telefragmentation of aluminum nanofilm by fourth plasmon excited by Q-switch pulse neodymium laser, second harmonic, of high intensity of SPR conditions was shown. The application of the pulse laser radiation allows the formation of transfers through air gap, 50 micrometer of the aluminum nanoparticles from aluminum thin film under self-plasmon resonance conditions at 532 nanometers instead of ultraviolet range 240 nanometers. Nanofragmentation process of the aluminum within film is caused due to interference of the plasmon polydipton subsystem stimulated by the lateral temperature gradients, microablation, subsequent transfer of the aluminum fragments through the air gap, and implantation into the polymer layer. The size of aluminum nanoparticles is in the range from 50 nanometers to 300 nanometers with a predominantly spherical-like shape. The next investigation of forced plasmon-assisted nanofragmentation aluminum thin film is planned in the direction of studying the process of aluminum nanoparticle transfer into the colloidal solutions and controllability on their sizes. Thank you for your attention.
Thank you a lot for your report and your research. And maybe some question from hall or from our online audience. One moment. It seems that we didn't have any question to you, but maybe one question from my side. Could you be so kind to share with us information where is possible to use your result of your research? How you think? What is the application of it?
As I told at the beginning, at the start, at the very beginning from in my report, the aluminum plasmonics, it is has a very attraction and attention for research. For example, for solar elements, for example, for medicine as particles, for so-called photothermal therapy, and many another application. Mainly it is interesting for solar cell and for medicine.
Okay, and as a rule for medicine, use noble metals, argon, silver, gold, something like this. You show us aluminium thin films.
Aluminium sorry, thank you. Aluminium has an interesting property, very high absorption coefficient in the windows of transparency, maximum transparency of the human body. At 800 nanometers near infrared. That allows to use this for heat by infrared laser, these aluminum particles, and increase the temperature needed for the therapy.
Thank you a lot.
About this, I have some, if you're interested, I have some literature reference and can send you.
Thank you a lot for your report and your answer. And now we are moving to our next speaker in this section.
Thank you very much.
It's kindly pleasure for me to invite. Can I check if Maxim with us?
Yes. Hello, dear colleagues. Can you hear me?
I cannot to see you. Yes. Now
You're right.
I think it's okay. Yes, yes, yes. You can start. Please take your floor.
Okay, thank you, first of all, for the opportunity to speak at our conference. I will be brief. Please tell me if I am overusing the dedicated time. Let me introduce you to our work on optimization of gold nanoparticles employed in hybridization by a sensor system based on surface plasma resonance. The aim of our research was to optimize DNA-bound gold nanoparticles for the purpose of their application in DNA hybridization biosensors and long-term storage in SSC buffer solutions. The SSC, or sodium saline citrate, is a commonly used buffer solution in hybridization sensor systems. The focus of this study was the phenomenon of surface plasma resonance, or SPR. This effect in towards is characteristic of a surface plasma, an electromagnetic wave that propagates along the boundary between a thin metal film and its external environment. The evanescent field of the laser beam is used to excite the surface plasma wave, as shown on the diagram on the left. The amount of energy subtracted from the light beam depends both on its angle of incidence and on the refractive index of the external environment, which in our case represents the mass of molecules absorbed on the gold sensor surface. On this side, you can see a schematic representation of the principles of operation of the traditional hybridization SPR biosensors, figures 1 through 3, and a SPR biosensor developed over the course of our work, figure 4. The difference between them is that the system we employed includes an additional amplification stage with the use of sandwich-type nanostructures consisting of immobilized probe oligonucleotides, target oligonucleotides from the studied sample, and gold nanoparticles. Here you can see the schematics of the modified nanoparticles, as well as the sandwich nanostructure based on the affinity interactions between its two intrinsic probes and one target molecule from the studied sample. This slide features the materials used in our work. I want to emphasize the absence of hybridization between the two probe oligonucleotides, which you can see on figure B. This lowers the probability of non-specific binding of gold nanoparticles to the sensor surface. Here you can see the results of experiments related to non-specific interactions between the biosensor surface and the gold nanoparticles. The graphs of different colors represent the levels of sensor signals after injection of non-complementary nanoparticles with different surface modifications. The nanoparticles that gave the smallest signal, which is represented by the gray graph, were chosen for further experiments. Here are the full results of experiments on non-specific interactions between sensor surface and gold nanoparticles. The less the signal, the better. Here you can see the response of the biosensor system to complementary interactions between the sensor surface, target DNA, and the modified gold nanoparticles, resulting in the assembly of sandwich nanostructures, which seriously amplifies the sensor signal, as you can see on this graph. This slide contains a calibration curve for a traditional hybridization SPR biosensor without gold nanoparticles, and a calibration curve for the nanoparticle containing biosensor system, with the latter exhibiting superior sensitivity and detection limit. Lastly, we determined the buffer solutions for the best functioning and storage of modified gold nanoparticles. In order to select the best environment for the long-term storage, a number of experiments was conducted. Their aim was to stimulate the aggregation of gold nanoparticles by reducing the distance between them through successive centrifugations. After each such centrifugation, the gold nanoparticle samples were resuspended with subsequent measurements of their optical density, as you can see on this graph. Their concentration calculated through the optical density was compared to the initial concentration, after which a conclusion was made about the number of gold nanoparticles that underwent irreversible aggregation and did not undergo resuspension. It was determined that the ideal stability of gold nanoparticles was observed in conditions of deionized water, as well as low concentrations of sodium saline citrate, such as 0.1 or 0.2 multiplicity. Allow me not to read out the conclusions, since they were formulated previously, but only to summarize that as a result of our research, the sensory characteristics of DNA-based SPR biosensors were improved, and the technology of modification and storage of the corresponding gold nanoparticles was developed. Thank you for your attention.
Dear colleagues, we still have time for one or two questions, if any. Yes, we have one question in the hall. One moment.
Thank you very much for this nice presentation. I wonder if I missed that, or did you mention any explanation of the reasons why you have such enhancement of your interaction between this evanescent wave and those nanoparticles of gold, which are like linked to the surface but not contacting it directly? And is it a wavelength dependent mechanism or not?
Thank you for your question. This is the fundamental question about our method, and in brief, I can explain that the amplification of sensor signal is attributed to two parameters of gold nanoparticles. First, their ability to excite localized surface plasmon waves, which draws energy from the incident light and enhances the signal that we detect on our spectrometer. And the second reason is the huge molecular mass of these gold nanoparticles. These were the nanoparticles in diameter of 13 nanometers, so I cannot recall their precise molecular mass, but it can be calculated, and it is much higher than the mass of the target DNA we detect. Did I answer your question?
Yeah, thank you. I think it's clear.
Thank you.
Thank you a lot for your presentation. And this section, which consisted from only 3 speakers, is closed now. And we have lunch break. And our next session will start at 2 p.m. So the session is closed. Thank you for your attention.
Dear colleagues, let's continue our work. Our next session called Nano-Object Microscopy and Nanostructured Surfaces. And the first speaker should be Dr. Mykola Danilenko from the Institute of Problem of Material Science of NASA of Ukraine. Dr. Danilenko, are you here? Okay, we can see you. Please share.
Okay. And now is it normal or not?
It's not a full screen mode.
What I have to do?
Please stop sharing the screen and then press again, share the screen and choose the right windows with full screen mode presentation.
Demonstration of screen. This presentation.
Okay, now it's good. Please start. Yeah, you can start your presentation.
Okay. The title of presentation: Structure and Properties of Coating made in of multi-component alloys. This work was carried out in the Institute of Problem of Material Science. My name is Danilenko Mykola, and some words about high entropy alloys. High entropy alloys are 20 years old, and they have attracted increasing attention because of their unique composition, microstructure, and adjustable properties. The first paper about multi-component alloys were published in by Inoue. He used this multi-component system for stabilization of amorphous alloys. Rangatanan called this multi-component system as metallic cocktails in 2003. Yeah, determined that the high entropy alloys contain at least 5 principal elements, and the concentration of each ranges from 5 to 35%. But the first multi-component alloys was obtained by student described in his master degree thesis in 1995. They expected to obtain multi-phase, multi-component alloys, but they produced one-phase alloy because high entropy for mixture for liquid state is very high, and this is reason of formation one-phase solution. It's well known that for two component, for example, titanium aluminum and titanium silicon, there are many chemical compounds and multi-phase, and to produce to obtain this one-phase alloys, they did not expect, and after that, it was proposed to divide the world of alloys into 3 types. Low entropy alloys. These alloys, traditional alloys, which know based on iron, aluminum, copper, molybdenum, tungsten, and others. These common alloys. Medium entropy alloys consist of 2 or 4 elements, principal elements, and high entropy 5 and more elements. We cannot say this situation. There were in this alloys there is no host elements. We cannot say that this alloy based on iron or other. In our institute, this authors publication that obtained some alloys consist of 26 elements. It's a record. This illustration example of some alloy. For example, we have, for example, chromium lattice, body BCC lattice, and when alloy some adding of vanadium, we see that some distorted because they have different radius, atomic radius, and serious distorted lattice caused by when multi-component alloys. Type of lattice depends on concentration of SD electron per atom. As a table, you can see that HCP, BCC, and FCC metal. High entropy alloys may be used as a construction materials, as electrode for welding, because and coatings, because some elements which are in the coating or electrode may be in welding parts and on substrate. It's good for producing such elements. In our case, sputtering targets were made from high entropy alloys in arc melting atmosphere of high purity argon. And the ingot was turned over and melted again 6-7 times for producing without homogeneous of elements. To obtain high entropy coatings, vacuum spray, arc spraying, and the method of ion sputter in compressed vacuum arc discharge plasma were used. This size of substrate and this surface of substrate before coating was special prepared. In this table, you can see results of mechanical characteristic of cast alloy as the initial state and the coatings. You can see that hardness of coating is higher than cast alloys. In the highest hardness demonstrates this alloy with this structure. And you can see that after in coating, the ratio between lava's phase and BCC changed. This caused by decreasing of some elements which have low melting temperature during deposition. In this case, you can see that BCC phase became from 24 became 71 because aluminium is decreasing of aluminum during deposition. Decrease of lattice parameter in high entropy coating by 10% leads to increasing of hardness and modulus, Young modulus. Some words about peculiarities of analysis of thin foil film by analytical transmission electron microscopy. This you can see that samples after grinding and analysis of this line for titanium aluminum interface. Such shape is typical for aluminum and titanium because they are mixed without any problems. It's well known in the intermediate zone is about 3 micrometers. My colleague from Kharkov Physics and Technology Institute, in order to connect stainless steel and titanium, used some intermediate layers, copper and niobium. And when they studied contact interface copper niobium, they published such results. Then this zone intermediate is about 3 micrometers too. But we know that niobium and copper don't mix. Thin foil was prepared for them, and we see this boundary, this interface, niobium copper, and scale, 100 nanometers. And analysis on line profile was received, and we see that intermediate zone is about 20 nanometers, comparable with 3 micrometers and 20 nanometers. This phenomenon we explain as following. When electron beam in scanning electron microscopy interact with bulk sample, we see this here trajectories of electron in sample. In case of thin foil in TEM, thickness is 100 nanometers, and beam diameter of beam is 0.7 nanometers. This area disappears, and we analyze only small piece of cylinder diameter is diameter with 0.7 nanometers. And high resolution picture of interface copper niobium, and this this picture is a fast Fourier transformation of selected area. You can see that scale 5 nanometers compare this with 3 micron. And this picture you can see the samples after bending with sample with crack between niobium copper, and fracture was in copper because contact copper niobium stronger than copper. And this picture represents the structural and chemical homogeneity of high entropy cast and coating. You can see the non-monotonous changing of concentration. Distance between points of analysis is 6 or 7 nanometers. And despite of such distribution of concentration of elements, the concentration S plus the electrons per atom is the same. It's nature, it's the main factors. And high resolution term of coating, after fast Fourier transformation, we detected some clusters. The size of clusters may be the same as for analysis in these graphs. Now, the result of analysis is presented as a table, and we will work with only yellow arrow. Let's.
Sorry, Dr. Danilenko, you should move to the conclusion because we have no time left.
Okay, let's arrange this all spectra in such a manner. Vanadium from minimum to maximum, and spectrum points of this number 9, here number 3, and here you can see the when tantalum is arranged from minimum to maximum. Such procedure was have been made for each elements for coating, and cast state, and we perceived such dependence. Despite of such characters of coatings, we can see that some zone with free of concentration. Such situation is forecast is more common, but and we can see that with consideration for gadolinium, some elements is increasing, but some elements are decreasing is for this. And but this result were made from 12 and 15 points. More detail about such effect, such development of result will be discussed maybe next time for two phase and one phase and dendritis in dendritis zone for such high entropy alleys. And conclusion. And thank you very much for attention.
Thank you very much for this very interesting report. We have time probably for one short question if we have. Do we have any question? We have no questions. So once again, thank you very much for your report. And we will move forward to our next speaker, Dr. Vitaly Belyanich from Uzhhorod National University, Ukraine. Dr. Vitaly, please take your floor.
My report is under the title, The Formation of Surface Nanostructures of Arsenicum sulfur chalcogenide films under electron beam irradiation. The chalcogenide glasses film are non-crystallized materials based on sulfur, selenium, and tellurium. And these chalcogenide films are disordered materials that have semiconductor properties. It is possible to obtain these materials with different structure, dimensional, and it is possible with a different dimension from 1 to 3 of the structure network, depending on the concentration of chalcogen in the glasses. In this figure shows chain and right elements of these glasses and films, and 2 dimensions and 3 dimensions structures. Glasses and films used in infrared technology, infrared and fiber optics, as well in other areas. Chalcogenide glasses as transparent in the infrared region. And these materials are transparent, is waveguide, then 11 micrometer, 15 micrometer, and 20 micrometer, respectively. Chalcogenide materials use photosensitive elements in devices. The chalcogenide films are used to record information on optical media to manufacture planar waveguide, microlenses, holograms, and photolithography. In this work, the electron-induced effects in arsenicum sulfur films were investigated. For studied this phenomenon, arsenicum sulfur films with arsenicum concentration 10, 15, 20, 25, 35, and 40 atomic percent arsenicum were used. Arsene Selen films were obtained by the method of resistive thermal evaporation in vacuum of bulk glasses onto sapphire substrates. The thickness of the films measured by the interferometric method was 4 micrometer. For investigation, we used electron microscopy with electron beam using the scanning electron microscopy model Vega, accelerating voltage equal 30 kilovolt, spot size 640 nanometer, and the electron beam current 60 nanoampere. We performed irradiation at the exposure time from 10 microsecond to 5 seconds. The film surface and the surface potential in the irradiation region were investigation using a scanning probe microscope, icon model, and Kelvin probe force microscopy measurements were performed at ambient condition. For each irradiation dose on surface of films, were formation matrix 20 by 20 irradiation points. The distance between the centers of the irradiation area was 10 micrometer. And in this figure, we can see a matrix of craters 20 by 20, and in this figure, we can see 20 by 20 craters of point irradiation area. Figure 8 shows the dependence of shape of surface relief of chalcogenide films, arsine sulfur, with different concentration and different irradiation time from from 100 microsecond to 1 second, and the cross-section of this surface relief. We can see that on the surface of this film formed relief on cone shape, crater shape, or combined relief formatted. And in low dose electron beam irradiation, those form relief in the cone shape, and in film arsenicum 15, selenium 85 formed sharp cone. This is Taylor cone, and he have a height 1 micrometer. Figure 9 shows the dependence of parameter of electron-induced relief of arsenicum selenium film, depth and height, and dynamic of change of this parameter. We can see that if irradiation time increases, this parameter change increases or decreases. If irradiation time less than 1 microsecond on surface film formed relief in form cone with Gaussian cross-section. And if irradiation time more than 10 millisecond, formed Taylor cone. In this figure, shows the dependence of parameter, dynamic of parameter of electron-induced surface relief, and form of this relief for film arsenicum 15, selenium NT5. In this film, in region time irradiation from 1 millisecond to 10 seconds, we can see the inverse of shape of this surface relief. And this figure shows the concentration dependencies of sensitivity, inverse dose, and maximum irradiation dose of these films. If atomic concentration of arsenicum in these films increases, then inversion dose increases, and sensitivity these films decreases. A high sensitivity have films with chemical composition, arsenicum arsenicum 25, selenium 75. This figure shows changes in surface potential and the surface potential of irradiation areas of films arsenicum 15, sulfur 85. We can see that at the irradiation time is 50 microsecond. Surface potential is less than 5 millivolt. And if irradiation time is 0.2 second, then surface potential changes from -5 to plus 40 millivolt. We developed two-layer charge model of electron-induced processes of chalcogenide films. When primary electrons penetrates into films, formed a two-charge layer. If electron beam penetrates into films, backscattering electrons and secondary electrons yield onto films, and near surface formed a thin layer, a positive charge in this area. We determined by the formula of Kanayo and Okayama the size of interaction region. For investigating film, this parameter is from 6.2 micrometer to 6.5 micrometer. We determined the depth of secondary electron yields, and for this film, this parameter is 4 nanometer, and the depth of reflected electron yield is 1.3 micrometer. This region is space charge region, and consists from two layers, positive charge layer and negative charge layer. This figure shows the evolution of induced by an electron beam surface relief in arsenicum 15 sulfur 85 film, depending of the irradiation dose and the charge of distribution in the irradiation region at energy of primary electrons 30 kilo electron volt. If internal electric field less more than critical density of charges, then in space charge region is collapsed and formed Taylor cone of surface these films.
Dr. Belanic, you have a little time left, so please quickly.
Conclusion. It was found that not monotonic surface structurings of the arsenicum sulfur films going upon irradiation with an electron microscope beam with an incident electron energy of 30 kilo electron volt. 3 interval of irradiation doses of this film were determined, in which are processes of charge accumulation and redistribution occur in the region of the interaction with the electron beam. It was established by the KPFM method that a positive charge is concentrated on the surface on the cones and a negative charge dominates in the craters. The dynamics of the formations of an electron-induced surface relief in arsenicum sulfur films is explained within the framework of a two-layer charge model. Parameters of this model have been determined. Thanks for attention.
Thank you very much for your presentation. Do you have any question? We have no questions. So once again, thank you. And the next speaker will be PhD student Mykola Sokolovsky from the Patton Electric Welding Institute of NASA of Ukraine. Mykola, can you hear?
Yes, I'm here. 1 second. Okay. Is the screen working properly? I see that it does. Greetings. My name is Mykola Sokolovsky from Patton Electric Welding Institute of National Academies of Science of Ukraine, and I am the presenter for this presentation on the influence of the substructure on the change of mechanical properties in the surface layers of structural steel during laser and laser plasma alloying. Durability and longevity of various parts and mechanisms, such as those presented on the slides, greatly rely on their operating conditions and above all on the methods of their hardening and alloying. Studies in this field, including promising new studies into new advanced processing technologies such as laser and laser plasma hardening and alloying methods, allow us to improve the quality of various alloyed surface layers. The usage of this approach allows the formation of surfaces characterized by a high level of hardness, thermal, wear, and corrosion resistance. For this reason, a study of the structure of alloyed layers depending on the processing modes, as well as the relationship between the structure and the properties and the causes of microcracks, is an urgent task for optimizing and correcting surface alloying technologies. This study is devoted to the study of surface alloying of steel and surface structures formed under different irradiation conditions in order to optimize the technological modes of laser and laser plasma alloying. Laser and laser plasma surface alloying was performed on the end surfaces of ring samples made of 38Hn3 MFA structural steel, the chemical composition of which you can see on the slide. For laser alloying and laser plasma alloying, a mechanical mixture of powders with a fracture of up to 40 microns with the compositions shown on the slide was used. The material processing modes for both technologies, as well as the mechanical characteristics of the steel as is, are shown on the slide as well. For the laser part of the alloying process, a Röfen-Sinar neodymium yttrialuminum granate laser from Germany with a wavelength of 1.06 nanometers was used. The equipment and technologies for these processes, namely a laboratory stand, a laser head, and a microplasma torch, were developed at the Patton Electric Welding Institute. The study of the samples was carried out using methods that include light microscopy, analytical scanning electron microscopy, and microdiffraction transmission electron microscopy. Utilizations of these methods allowed us to fully analyze the structure of the alloyed layers from multiple perspectives, creating a more complete data set. During laser alloying with working mode shown on the slide, the structure formed in the treated surface layers is characterized by the increased microhardness relative to the base metal. Cracks of up to 500 nanometers were observed in the alloyed layer and in the fusion line area. It has been established that the tendency to crack under this mode of laser alloying is happening due to the structural and concentrational changes associated with the redistribution of elements, in particular chromium. Studies of the presence and distribution of particles of phase formations of different compositions has shown that a relatively uniform distribution of tungsten carbides, size of up to 25 nanometers, is observed in the internal volumes of the grains of the alloyed layers. The formation of clearly visible local zones of increased chromium concentration, up to 25%, is observed along the grain boundaries. Studies of the microstructure have shown that the laser alloying for a sample results in the formation of intercrystalline needle structure in the surfaces of the alloyed layer, saturated with extended carbides of various sizes along the intergradual boundaries. In this case, the microstructure is characterized by a sharp increase in density of the dislocations along grain boundaries to values that significantly exceed the density of intergranular dislocations. Such long and dense accumulations of dislocations are associated with a high chromium content in the boundary zone and the formation of chromium container carbide faces at grain boundaries. Significant changes in the structural and phase state of the alloyed surfaces are observed under the laser alloying working mode shown on the slide. This working mode is considered optimal for this metal. It is characterized by the formation of a fine-grained martensitic austenitic structure with a significant decrease in the grain shape factor. This indicates that under optimal laser processing condition, the surface structure is fine-grained and evenly balanced. The distribution of chemical elements is also relatively uniform. The tungsten content in the grain volume is 6 to 7%, rising to 9% in boundary zones, while the chromium content is homogeneous at 12 to 16%. This indicates that there is no gradient in the distribution of chemical elements in the surface layers of the metal. The surface layer have a minimal number of pores, no cracks, and a slight change in microhardness in the heat affected zone. This laser alloying working mode is characterized by a uniform distribution of dislocations with a general decrease in their density. The sizes of phase separations in the surface layers are more dispersed and evenly distributed in the internal volumes of the grains. After laser plasma alloying at 3 kilowatts with overlapping of 4 tracks, a crystalline type structure is formed in the treated alloyed surface layers, but with grain enlargement and changes in their shape coefficient. A rather high porosity was found, as well as large tungsten carbide inclusions of up to 230 nanometers in size and up to 10% quantity. In addition, rather significant changes of chromium concentration in certain structural zones were identified, with an increase to up to 17% or more of chromium being observed mainly along the intergranular boundaries and diffusion line. This working mode is also characterized by a clear localization of crack formation along the fusion line. The largest chromium concentration gradient is also observed. The study of the fine microstructure showed that under the condition of laser plasma alloying in this sample, the structure of the metal of the surface layer is characterized by the formation of a needle structure with a large number of phase separation caused by carbides. and with a sharp gradient in the density of dislocations along large phase separations. At a different working mode, where only one track is applied, the one that we consider optimal, a slight increase in the hardness of the surface layer is observed, but without the gradients present in the base metal. Significant dispersion of the structure of the martensite, carbide faces, and its inclusions, uniform distributions, both in volume and along grain boundaries, of chemical elements, such as chromium and tungsten, as well as the complete absence of porosity and cracks. The study of the microstructure revealed a general decrease in the density of dislocations and the uniform distributions in the volume of the surface layer during laser plasma alloying of this sample. The dislocation substructure is characterized by a cellular type with a cell size and the presence of dispersed phase in the internal volumes in the grain. On the basis of these experimental studies, an analytical assessment of the differentiated contribution of the structural and phase parameters to the change in the technical characteristics of surface layers, as well as the distribution of and level of local internal stresses in the treated surfaces and their gradients was performed. Analytical estimates of the integral value of material strengthenings, sum of sigma t, were made according to the well-known dependence shown on the slide. It has been established that under optimal conditions, an increase in the overall level of hardening was observed. This has to do with the dispersion and substructural hardening. The increasing role of dispersion hardening is related to the maximum contribution of carbide phase particles. At the optimum mode of laser plasma alloying, with the superposition of a single track, the maximum contribution to the increase of the material strengthening value is provided by substructural grinding at 176 megapascal and the effect of phase separations at 196 megapascal of both dispersed and large particles. It should be noted that the increase in the level of dispersion hardening during plasma alloying is largely contributed to the supply of dispersed phase and to a lesser extent to the large tungsten carbide inclusions. The results of the research at all structural levels also allowed us to estimate the crack resistance of the alloyed layers of surfaces treated in different modes. The analysis of crack resistance was carried out considering the distribution of dislocation density based on the assessment of the level of the local internal stresses according to the dependence shown on the table, on the slide. The study has shown that the surface layers formed under laser alloying conditions at 4.4 kilowatts with a speed of 400 millimeters per minute are characterized by a sharp increase in the density of dislocations. The presence of such zones in the surface layer is the basis of formation of sharp gradients of the local internal stresses. The fracture, or also known as the fracture concentrators. Their maximum values are 18,500 megapascal. Laser alloying under the optimal mode of power of 3.5 kilowatts and an increase in speed to 500 millimeters per minute results in a reduction and the uniform distribution of the dislocation density. This ensures a minimal and gradient-free level of internal stresses of up to 70 megapascals in the alloyed layer. At the same time, the formation of crack nucleation and propagation zones was not observed. A rapid gradient in the distribution of dislocation density in the volume of crystalline particles was observed in the samples after laser plasma alloying with 4 overlapping tracks. The increase in chromium concentration, up to 17%, is clearly visible and is the main reason for the formation of elongated internal stress concentrators, that is, crack nucleation sites, where local internal stresses vary from 300 to 14,000 megapascals. In the case of laser plasma alloying at the optimal mode with a single track overlaid, the presence of discretely sized, evenly distributed phase separations in the grain volume, accompanied by a low dislocation density without gradients, was observed. This indicates the absence of structural conditions for the formation of internal stress concentrators. At the same time, the local internal stresses values decreased to up to 370 megapascals, which characterizes the structural state of the surface as optimal and confirms the practical absence of cracks. After conducting a wide range of studies, we can conclude that experimental and analytical studies have established that the influence of the structural phase state of the metal and alloyed layers on 38HN3 MFA structural steel formed under different modes of hardening and alloying treatment on the change in the strength and crack resistance characteristics of the treated surfaces was performed by laser and laser plasma alloying with a tungsten carbide chromium powder mixture. It was shown that the modes of laser plasma alloying contribute to an increase in surface hardening on average by 20% compared to just laser alloying, with the greatest contribution to the hardening at the optimal mode being phase separation and substructural. Laser plasma alloying using optimal modes ensures maximum crack resistance of the alloyed layers due to the absence of crack nucleation and propagation zones caused by the uniform distribution of dislocations and density without any rapid gradients. Laser plasma alloying contributes to a uniform and gradient-free distributions of dislocation density in the absence of structural conditions for the formation of internal stress concentrators, that is, crack nucleation and propagation zones. Crack formation and after laser plasma hardening of alloyed layers of structural steel is usually associated with the formation of dense and elongated dislocation clusters, mainly along intergranular boundaries, which is facilitated by the uneven distribution of chemical elements such as chromium. If you have any questions, feel free to ask them. This presentation is now complete.
Okay, do we have any question? No question. So once again, thank you very much. And the next speaker should be PhD student Mykola Sidorenko from the Gas Institute of NASA of Ukraine.
One second, please.
Okay, we can see your presentation in full screen mode, so please.
Perfect. Greetings, my name is Mykola Sidorenko from Gas Institute of the National Academy of Sciences of Ukraine, and I would like to present to your attention a report on the topic creation of heat transfer coatings on heat-loaded surfaces to intensify their cooling. First, we must determine the feasibility of conducting such research. Based on previous research, we determined the following, that the boiling crisis is a value that reflects the maximum heat transfer coefficient, acronym HTC, possible under the given conditions for a specific heat carrier. For the nanofluid presented is in the graphs above, accordingly to reference 1, the HTC value is 200% on that for a base liquid distilled water. And this allows us to draw a conclusion about the expediency of the experiments presented in the report. The purpose of the experiment was to apply deposit of nanoparticles of the surface of a steel cylinder by the quenching method and to determine the stability of the applied deposit on the surface of the heat exchanger. The deposition process consisted in preheating a steel cylinder in a vertical resistance tube furnace to a temperature of 620 to 650 degrees by Celsius, followed by its cooling in a container with a cooling liquid, distilled water, nanofluids, with the possibility of adjusting the temperature. The conducted studies were performed at a coolant temperature of 99 to 100 degrees by Celsius. The following types of nanofluid were used as coolants for quenching. It's nanofluid NF8A, titanium dioxide base, AlC7A, aluminosilicates, montmorillonite and attapulgite, and NF21, carbon nanotubes based. In the process of cooling the steel cylinder in the
In in the nanofluids, nanoparticles dispersed in the volume of nanofluid were deposited on the heat exchange surface, thereby providing modification of a heat exchange surface. The sediment was mostly actively formed during deposition in NF8A, titanium dioxide-based nanofluid. The worst is NF21 carbon nanofluids-based nanofluid, which is shown in the photos presented below. The sediment was precipitated by sequential application of nanoparticles in 5 consecutive experiments. After the first experiment, cylinder was deposited sediment was again heated and cooled in the nanofluid. The result was additional deposition of nanoparticles on the heat exchange surface. Experiment continued up to 5 layers of sediment on the surface of the cylinder. Next, an experiment was conducted with the cooling of a cylinder with a multi-layered sediment in distilled water. As a result, cooling curves were obtained, which are presented in the graphs below. The presented photos show a layer of deposited alumosilicate nanoparticles on the surface of a steel cylinder after quenching in nanofluid AlC7A, aluminosilicate nanofluid. The application process was performed 5 times. The presented photos show a layer of deposited aluminosilicates nanoparticles on the surface of a steel cylinder after cooling in distilled water. From the summary of graphs of the cooling curves, it is possible to see the improvement of the heat transfer characteristics of the cylinder surface. The inconsistency of the initial temperature of the cylinder at the beginning of cooling did not affect the purpose of the study, namely the formation of a deposit on a surface with subsequent cooling in distilled water. This is true for further experiments. The presented photos show a layer of deposited titanium O2 nanoparticles on the surface of a steel cylinder. The application process was performed 5 times. The presented photos show a layer of deposited titanium dioxide nanoparticles on the surface of a steel cylinder after cooling in distilled water. From the summary of graphs of the cooling curves, it is possible to see the improvement of the heat transfer characteristics on the cylinder surface. Next, the presented photos show a layer of deposited carbon nanotubes on the surface of a steel cylinder. The application process was performed 5 times. The presented photos show a layer of deposited carbon nanotubes on the surface of a steel cylinder after cooling in distilled water. From the summary of graphs of the cooling curves, you can see almost identical characteristics of the heat transfer of the not the heat transfer cooling speed as more of the cylinder's surface. And the final stage of the research consisted in cooling a steel cylinder with deposited nanoparticles in boiling distilled water. The presented graphs show the cooling curves of a steel cylinder with applied nanoparticles during boiling in a nanofluid. Those nanofluids was titanium dioxide-based, aluminosilicates-based, and carbon nanotubes-based. Cooling was carried out in boiling approximately 99 degrees Celsius distilled water. As can be seen from the graphs, the most effective coating was formed by nanofluid AlC7A, aluminosilicate, montmorillonite, and attapulgite-based. This can be explained by the greater anisometricity of the nanoparticles. Conclusions. Formation of sediment on the heat exchange surface occurs very quickly. It takes literally a few seconds to form a layer with improved heat transfer. Second, since cooling occurs quickly in all temperature ranges, this means that nanoparticles distributed in the liquid and the ball layer also have a significant effect of the intensification of heat exchange. Anisometric parts are the most promising from the point of view of modification of heat exchange surfaces. The necessary thickness of the sediment, which improves heat exchange, is formed at the initial stage of cooling, and further modification does not play a significant role. The greatest effect is observed between the first and second immersion in nanofluid. The stability of the formed layer requires further research. Work reference to works previously published. And thank you for your attention.
Thank you for your presentation. Any questions?
May I have a question?
Yes, please.
Can you tell please about the thermal conductivity of your materials which are used for deposition on the surface of this tube? I mean, the mechanism of cooling is due to the different thermal conductivity, if I correctly understood your presentation.
Almost the main like force to enhance heat transfer is produce more boiling points on the surface of heat exchanger due to porous structure of nanoparticle sediment on the surface of heat exchanger. We know there's a boiling bubble of steam that when it's when it's came out of surface of heat exchange, it's take a part of overheated liquid from the surface of heat exchanger, and it allows to come more cooler liquid to take out the heat from a surface. And our experiments showed that porous like coat when occurs in second boiling crisis on heat exchange surfaces, it's a great resistance to take out heat over heater, and we must destroy this porous coat and allow the coolant to come directly to a heat exchange surface. And this sediment allows that to happen.
Thank you for your question and for your presentation again. And the next speaker, PhD student Ruslan Krivoshapka from National Science Center Kharkov Institute of Physics and Technology of the NES of Ukraine.
Can you hear me?
Yes.
I have some issues with my camera. I can switch on another device if it required, or can report immediately.
You can report without camera.
Thank you. Can you see my screen now?
Yes, you can start your presentation.
Okay. My name is Ruslan Krivoshapka. I'm a researcher of National Science Center Kharkiv Institute of Physics and Technology. And today I want to tell a few words about part of our work, which can be called as dependence on physical mechanical properties of chromium nitride / niobium nitride coatings on the deposition parameters. In the recent years, the development of vacuum arc coatings is based on the combination of coating components with different properties and the complication of the coating structure. Nitrides of transition metals are often used as components of a multi-layer system. Chromium nitride has good wear and corrosion resistance, high temperature stability, and a lower friction coefficient. In turn, niobium nitride has high hardness and electrical conductivity, good heat resistance, and chemical inertness. And when these two materials are combined, it becomes possible to obtain new properties and expand the field of their application. So this report presents the result of studies of the influence of deposition technological parameters on the physical and mechanical properties of vacuum arc multi-layer coatings based on chromium and niobium. So vacuum arc nitride multi-layer coatings based on chromium and niobium were deposited in a modified installation, blood 6. The evaporator materials were pure metals of vacuum melting, chromium and niobium. Stainless steel samples with a mirror polished surface were used as a substrates for deposition of coatings. They were used to determine the properties of coatings. Previously, the samples were washed, and during the deposition, the samples were placed on both sides on a vertically located metal substrate holder in the vacuum arc chamber of the installation. Substrate holder was in the form of a rectangle. It was placed on the same horizontal level as the evaporators and rotated around its vertical axis. Before deposition, the vacuum chamber with the samples was pumped. After this, the ionic cleaning and activation of the sample surface were carried out by bombardment with metal ions with continuous rotation of the substrate holder and a constant voltage on it. The process of sample cleaning and heating lasts for 10 minutes. After these processes, an underlayer of pure metal, chromium and niobium was deposited to improve the adhesive properties of the coatings. The technological process of deposition took place within 1.5 hours, and the pressure of the reaction gas in the vacuum chamber varied from 0.08 to 0.27 pascals. The constant negative voltage on the substrate was minus 70 volts and minus 200 volts. The arc current of evaporated was 80 ampere for chromium and 100 ampere for niobium. The evaporators had plasma flow focusing by a magnetic field. The distance from the evaporators to the axis of substrate holder rotation was 500 millimeters. So 3 series of coatings were obtained with continuous rotation of the substrate holder, series 1, at the interval of the substrate holder rotation of 80 seconds, series 2, and the interval of 20 seconds, series 3. Planar raster electron microscopic images were obtained, and the samples were examined without applying any coatings. Cross sections were examined in a field emission scanning electron microscope. The study of physical and mechanical characteristics was carried out by the microindentation method using CMS hardness measurement equipped by standard Vickers pyramid. A microtester equipped by a trihedral Berkovich pyramid was used to determine Young's modulus according to the test method by Oliver and Farr. Technological parameters of coatings are current of evaporators, constant negative voltage on the substrate, pressure of reaction gas are presented in table 1. 3 series of coatings were obtained by using of different technological modes. Coatings with different thicknesses of individual chromium nitride and niobium nitride layers, and bilayer period, number of layers 68 and 2070 layers, and kind of the interface between the layers, clear boundary or transition layer. Figure 3 demonstrates some images for the surface morphology of 3 series obtained and the same technological conditions. The turning off the power sources of the evaporators for this time of turning the substrate holder affects the processes occurring on the cassette's surface and in the near cassette zone, which in turn further affects the deposition processes of the vacuum arc coating, the structural phase state, and the physical and mechanical properties of the coatings. Figure 4 demonstrates some images for the surface morphology of third series obtained under different technological conditions of deposition. Figure 5 demonstrates some images of cross-section of coatings with different thicknesses of individual chromium nitride and niobium nitride layers, and by layer period, the number of layers and the kind of interface between the layers. So the boundaries between the layers are clearly visible on figure. Analysis of the cross-section showed that the total thickness of the coatings was 3.6 micrometers for the first series and about 8 and 9 micrometers for the second and third series respectively. Table 2 shows the geometric parameters of the coatings and evaluation of the thickness of individual chromium nitride layer and individual niobium nitride layer for coatings of the first series was not carried out, since these coatings do not have clear transition boundaries between layers due to the formation of transition layer chromium, niobium, and nitrogen. The microhardness of the studied coating is presented in table 3 in the form of the minimum and maximum values of the microhardness, the range of the obtained values, the average value, and evaluation of the microhardness was carried out by 8 measurements at a distance of 100 micrometers between them, with a total measurement length of 0.7 millimeters. The analysis of the average microhardness value showed trends in change in the microhardness of coatings depending on change in technological parameters. Increasing the constant negative voltage on the substrate leads to the reduction of the average microhardness of the first series, and increase in the average microhardness of the second series, and the preservation of the microhardness of the third series. An increase in the pressure of nitrogen in the vacuum chamber leads to an increase microhardness of the first series, and the decrease of the second and third series respectively. The dependence of the average microhardness value of the number of layers is presented in figure, and an increase in the number of layers from 68 to 270 leads to an increase in the average microhardness value by 4 to 39% at various deposition parameters except one mode, when average microhardness decreased by 8.5%. To evaluate the heterogeneity of the structural phase state, we can use the spread of the obtained microhardness values of the coatings and root mean square deviation. An increase in the voltage on the substrate leads to a decrease in the spread of microhardness values, all 3 series. This indicates that the obtained values cluster around the average value of microhardness. Thus, when the voltage on the substrate is increased, coatings with a more uniform structure are formed. An increase in the pressure of the reaction gas in the vacuum chamber with constant voltage leads to a decrease in the spread of microhardness values of coatings of the first series, and increase in the spread of microhardness of the second and third series. Table 5 presents the average values of the elasticity modulus, the hardness medium to elasticity contact modulus ratio, which characterizes the structural state of coatings and the property of the material to resist mechanical impact, and the hardness medium cubed to elasticity contact modulus squared ratio, which characterizes the materials resistant to plastic deformation. So The investigation of vacuum arc multilayer nitride coatings obtained under different technological parameters of deposition made it possible to expand its influence on the physical and mechanical properties of coating. It was obtained 3 series of coatings with different thicknesses of individual layers and bilayer period, number of layers and nature on the interface between the layers. The total thickness of coatings was 3.6 micrometers for the first series and about 8 and 9 micrometers for second and third series respectively. For the third series, the preservation of the average value of microhardness is observed when the nitrogen pressure and negative voltage on the substrate change. An increase in the number of layers from 68 to 270 leads to an increase in the average value of microhardness by 4 to 39%, except one more, when it decreased by 8.5%. This change are associated with an increase in the number of interface boundaries, which affect the processes of the formation of individual layers and diffusion between them. This leads to increased crack propagation resistance and elastic properties of coating. An increase in voltage on the substrate from minus 70 to minus 200 volts at a constant pressure of the reaction gas leads to a decrease in the spread of microhardness values of all 3 series. So this indicates on the fact that the obtained values cluster around the average value of microhardness. And during increasing the number of layers in the coating from 68 to 270, the elasticity contact modulus decreased by 6 to 8% at a voltage minus 200 and increased by 8 to 25% at a voltage minus 70 volt at various pressures of the reaction gas. Now my report is over. Thank you for your attention.
Thank you very much for the interesting presentation. Do we have any question? No question yet. So it was the last talk for today. And I give the floor for the head of the organizing committee, Fesenko Elena.
Dear colleagues, let me to start from congratulation on the most significant and momentous event in our nation's history, Independence Day of Ukraine. Everything in our life has started and everything in our life has end. Taking into account it, our conference NANO-2024 is going to the end too. Let's together remind some moments and statistics of the conference. This year, our conference took place at National University. And I want to thank the rector, Professor Vladimir Smolyanka, and all staff members of National University for help us in organizing this conference. The conference was opened by Maria Cristina Russo from Directorate General for Research and Innovation from European Commission. Also, welcome speech was done by Boris Mozolevich, Director General of Science Development Directorate of Ministry of Education and Science, Director of National University, and me as organizing committee. This year, our NANO conference consists from oral workshop and three special sessions. For example, the role of science and innovation in the course of the recovery. And the second session, the programs and instruments for science and innovation. important that we had quite discussion during this special session, the role of science and innovation in support of recovery in Ukraine, with the representative of European Commission, with representative of National Academy of Science, President of National Academy of Science Zagorodny, and also Ministry of Education and Science of Ukraine. It's a pleasure that we already received some warm feedback from our participants and active promotion of our event. Namely, our events was promoted on website and social networks of Ministry of Science, Education of Ukraine and the National Academy of Science of Ukraine. Also, I ask all of you, you can share any video, photos, and poster which is posted in our social network. I hope that each of you know our website and Facebook and LinkedIn page of our conference. Maybe you remember that our conference was initiated in the frame of FP7 EU Nanotwinning project in 2012, and it's a nice example how the outcomes of EU project can continue its own life after the end of the project. The project finished many years ago, but the Nano conference continued its life for last 12 years, and now it became the biggest scientific conference which held in Ukraine. Each year, our conference has more than 500 participants from mainly countries. We prepare each year extra food for Nano conference, and as a rule, it's more than 800 pages. So it's quite hard work, and it took a lot of our time. Also, we prepared special review in international journals and a special series of interviews. I think it is total, we have published more than 2,000 articles, more than 12 special interviews, prepared about special international jobs. Also, we try to support technology transfer and development of innovation products, international cooperation and close cooperation between research, developers, innovators, and engineers. Each year, we try to involve researchers and we try to invite innovation companies. We have also representatives of England Innovation Company, which produce equipment. And in the future, I plan to more active do it. And also, sometimes we organize online matchmaking event, and I hope that our conference is something like platform for communication, for looking new partners, and in the future, maybe you will work together, maybe you will apply to some programs to prepare some common projects, and maybe we'll develop some developers. It's also important that the COVID and the war didn't stop us. From the start of the war, we started holding Nano conference in hybrid format. When in the 2022 year, the first year of the war, we organized the Nano 2022 in hybrid format. It's a lot that we can see that. Then in the first year of the war, it was about 150 and about 550 online. This year, our despite the war, organizing committee of Nano 2024 received more than 600 safety application forms from participants from different countries. In total, during 4 days of conference, we had about 10 plenaries, invited lecture, and about 100 oral presentation, and about 350 poster presentation. Please pay attention that all participants who sent us their poster in advance We posted in our Facebook pages of our conference in Facebook and LinkedIn in social networks. And also we created a special session on our website, poster, and you can find all poster there. So it's also additional our efforts because we wanted to help you disseminate information about your research as wide as it's possible. Please pay attention that taking into account that our conference was in hybrid format and we had live broadcast of each session of conference and each day you can watch a record of it in any time after the end of conference too. Also, we plan as usual to prepare a special issue in Springer book and special issue in molecular crystal, liquid crystal. Also this year we will prepare a special issue in new journals and I will inform you a little bit later in coming days. And I wanted to tell very big thanks to all my team because you know that such big event organized by team, not by one person, because it is not possible. So thank you a lot, each of you. And for all participants of NANO conference, I want to wish all the best and see you next year. Also, I sincerely thanks our defenders for their invaluable efforts, for the opportunity for us to live and to continue to work on our own land. We all wish their safe return to their families. I wish victory for our country and happy future in free, independent Ukraine. Thank you a lot. See you soon next year.